Welcome, everyone. We're excited for today's webinar, Synthetic Cells at the Frontier, Navigating Innovation and Risk. This webinar is being hosted by the National Academy's Committee on the Environmental, Biosafety, and Biosecurity Considerations for Synthetic Cell Research and Development, and will support the study committee's information gathering process and feature expert discussions to inform its final conclusions and recommendations. My name is Felicia Wu, and I'm the John Hanna Distinguished Professor of Food Safety, Toxicology, and Risk Assessment at Michigan State University. And I'm pleased to be a co-chair alongside Dr. Peter Carr, who is a senior scientist at RTX BBN Technologies, working at the nexus of synthetic biology and national security. We're lucky to have a wonderful group of experts as part of our study committee, many of whom are participating in today's webinar. Their full biographies can be found on our project webpage. And that will be on the next slide. This is the list of our committee.
Sorry. Hello, everyone, and welcome. As Felicia said, I'm Peter Carr. On the next slide is our committee's statement of task. Wait for that to come up one second. Briefly, this study is assessing both existing and needed frameworks for responsibly managing risks associated with synthetic cells, both for contained and environmental release applications, and doing so with the goal of informing future policy and safeguarding public well-being. On the next slide, just before we get started, we have a few reminders. Comments made during the meeting today should be attributed to individual speakers and not their organizations unless otherwise stated. In addition, any thoughts shared during this meeting should not be interpreted as the opinion of the National Academies. And the meeting is being live streamed and recorded for later use by the committee. And finally, please be respectful of all participants and speakers. And now I'll hand off to the moderator for our first session, Dr. Cameron Kim from Duke University.
Thank you, Peter and Felicia, for your introductions and beginning our symposium. My name is Professor Cameron Kim, and I am an assistant professor of the practice of biomedical engineering and the Science and Society Initiative at Duke University. And I am a member of the National Academy's Committee on Environmental, Biosafety, Biosecurity Considerations for Synthetic Cell Research and Development. And I'll be moderating today's first session, Frontiers of Synthetic Cells, Applications, Insights, and Implications. Thank you to the panelists who have volunteered to present, and thank you to those tuning in online to watch this session. This session will examine the cutting-edge research and development of synthetic cells across a wide range of applications, including biomedicine, biomanufacturing, biomining, and bioremediation. It will also delve into their use in probing the origins of life, exploring mirror biology, advancing fundamental biological understanding, and enabling space-based applications. The discussion will assess the current state of science in each domain, highlight the advantages and limitations of synthetic cells compared to conventional engineered organisms, and consider the associated biosafety, biosecurity, and environmental implications. Today, we have a great lineup of 8 speakers. To make sure we have plenty of time for discussion at the end of the talks, we will save any questions for the panel discussion. Each speaker has up to 7 minutes, and to ensure we have time for everyone, we will be keeping to this timing. I'll also keep introductions brief, and the speaker biographies can be found on the event webpage. In the first session, we will hear from Dr. Lynn Rothschild at the NASA Ames Research Center, John Glass at the J. Craig Venter Institute, George Church from Harvard University, Elizabeth Strahalski from the National Institute of Standards and Technology. We will then take a short break before returning for our second portion of the session, where we will hear from Jiming Tan at the University of California at Davis, Michael Booth at University College London, Neha Kamat at Northwestern University, and Juan Perez Mercader at Harvard. To begin, we will hear from Dr. Lynn Rothschild, Senior Research Scientist in the Planetary Systems Branch at NASA Ames Research Center.
The sharing is not coming up.
Give me one moment.
Ah, there you go. Okay. Okay, that should be fine. So thank you very much for inviting me. I am going to primarily talk about NASA's needs for synthetic cells, but was asked to give a short introduction. And what I'm really going to do is highlight a paper that many of us were on that came out last year, building synthetic cells from the technology infrastructure to cellular entities. And so I'll just sort of dance through that because you certainly can read the whole paper yourself. It's great bedtime reading. Now, when you're talking about what is life and synthetic cells, you really have to talk about both. You can't talk about synthetic cells if you don't know what the goal is. And if the goal is really to produce life, you have to be able to define life. And so in the paper, we used Tibor Ganti's model, where their primary properties of compartmentalization, replication, metabolism, that then flow into emergent properties of evolution, responsiveness, and movement. We use that for convenience. I actually prefer a little bit more philosophical approach to this. And again, we could talk about this for hours. And so this is clearly highly abbreviated. But in summary, I believe that life is an emergent property based on physical components. It's not just an object. And what we mean by emergence in philosophy of science is that you can't predict what's going to happen, even if you know everything about the components. For example, you could know everything about sodium and chloride, and that wouldn't predict what salt tastes like. Second, it's a population level phenomenon, so it may not be good enough to create a single individual. So for example, we know that life is self-replicating, but not every organism necessarily has to self-replicate, but as a population level phenomenon, it needs to. The process includes fighting entropy, and as a biologist, I hate to give the last word to Schrodinger, but I think he was really onto something, creating disequilibria, atmospheric, isotopic chirality, and that sort of leads into the mirror life to some extent. Physical components, at least in our solar system, I believe, are based on organic carbon chemistry and need a solvent, likely water, but synthetic cells may prove me wrong on that, and also needing energy and time. And so I'll just sort of finish with another philosophical point to hammer this home. When Da Vinci was asked to define water, there was a very long definition. Water is sometimes sharp, sometimes strong, sometimes acid, sometimes bitter. I won't read the whole thing, but you get the idea. It's a description of the properties of water. Now, if you are asked what water is, there's a very simple definition. Water is H2O. And the question that I pose for you is there actually a natural definition? Is there an H2O definition for life? Or at the end, are we still in a stage where there's a Da Vinci sort of definition where you're describing it? And even worse, maybe there is no H2O definition for life. And so maybe this is a fool's errand to try to find one. But again, I wanted to bring that into the thought process. So let me get back into the science in the paper a little bit. This is another one of the figures from the paper. We identified 4 different platforms for studying cellular processes, going from cell-free systems to encapsulated cell-free systems to this thing that we call synthetic cells, to natural cells. For those of you who don't recognize Darwin, that's him before he had 8 kids. And we also identified 3 different approaches to making minimal cells. There's the bottom up. There's, taking the ribosomes and the amino acids and everything else and shoving it in a vesicle. There's a top down approach where you take a perfectly competent cell and start pulling its guts out, or a middle out where you can sort of do a Franken-cell sort of thing. You're taking large numbers of components and putting it maybe into a simplified cell or a vesicle. And again, this is all in the paper. And finally, this is also a figure out of the paper, and I won't even delve into it, but this way you have it in the nice color, larger version. And again, I encourage you to take a look at the paper before you write your report. So that's sort of the quick intro. And many of the people speaking after me will be talking about aspects of that, and they will certainly also be delving in more depth into aspects of what I'm going to talk about for space applications. I could easily have been the last person and said, to echo all these other people, but instead I know that other people will be going into greater depth and do a much better job probably than I on many of these aspects. So I remind you, I work for the National Aeronautics and Space Administration, and I point that out because many people forget that aeronautics is our first A, and I do believe that synthetic cells could, in principle, help with green aviation, even though the words green aviation have been recently scrubbed from our websites. There's a potential for creating biofuels and certainly novel materials. But with that, I'm going to jump into the two areas that I really see synthetic cells as being much more important. One is the exploration issues, particularly as we're pivoting back towards human exploration. One of the biggest issues in exploration is upmass, and that is going against Earth's gravity well. So anytime that we can lower our upmass, we increase our mission capability. Second of all, we really need to have good reliability in extreme environments, and I'll show you what I mean then why synthetic cells can be applied to solving both those problems. And then, of course, we have a big science program, and that includes astrobiology, our questions of where we come from, where we're going, and are we alone in the universe? And for these big sorts of science questions, we can't really rerun the origin and evolution of life. We can't just set the clock back to the early Earth and try it again. And again, this is where synthetic cells can certainly help us solve our problems. So let's go back to the exploration bit. Life is a self-replicating micromachine that does carbon chemistry and nanotechnology. And I think that that gives you a good starting point to get an idea of what life can do for us in the agency and what can do for synthetic cells. Life naturally evolved will not necessarily suffice because, again, we have the upmass problem, we have the problem of extreme environments, and so on. Our lab uses life, what I call life 1.5, and it's really a synthetic biology approach. So we're really, in the end of the day, tweaking naturally evolved organisms with sequences that may be evolved elsewhere or completely new capabilities. But perhaps the solution really is making that next jump to life 2.0, in other words, synthetic cells. So I know we were supposed to talk about some of the things we will do. I'm involved in a lot of projects in my lab. My job the last 20 years has really been to look to see how synthetic biology can enable NASA's missions. So we have everything from chemistry, and remind you that life does chemistry on the Earth. There's no reason not to do it off planet. We have a big project going on in developing an astropharmacy, doing nanotechnology work using DNA as a building material rather than DNA as a coding material. Toyed a little bit with generating electricity, which of course all organisms can do. Material production, for example, fungal biocomposites for building in space, and that we're very heavily into right now, as well as biomining. Just got awarded a lot of money for detoxifying lunar and Martian water. On the moon, the big problem is mercury. On Mars, it's perchlorates. Food, flavors, scents are sort of on the horizon. But all this so far, except for the nanotechnology, has been done with really life 1.5, but I believe life 2.0 can really revolutionize that even more. And to hammer that point, here is something that's really trivial to everyone, I'm sure, listening in. At this point, we take digital information. When we're making DNA, we send it to a DNA synthesis site, and it doesn't matter whether it's across campus or on another planet, but we are using that digital information to then have someone else read it and create biological information. And this is going to not only revolutionizes how we do things on the Earth, but again, imagine that you don't need to take those sequences with you. You could make them on the moon or Mars. And once you're dealing with digital information, it is completely massless. So now, and thanks to Elizabeth for pushing me on this last night, what about sending the digital information makes the cells all together? So you don't need to take something with you, or you can pivot if you get to Mars and there's a solar particle event and you need to make something completely different that has been discovered elsewhere. So I've rather taken now with the idea that the ability to make cells from scratch was really the ultimate in massless exploration for us. Finally, on the science side, and this is my last pivot, synthetic cells allows us to answer the what if questions. What if it had happened differently? Is there any reason that we have left-handed amino acids and right-handed sugars? In other words, could we have had a mirror life form? Is it just because there was an enantiomeric excess in the meteorites and the abiotic settings that we drew on for the origin of life? Or is there a good reason? What if you did something completely differently? And this is really, really important for us in astrobiology because of the imperative that we have to search for life elsewhere that goes from looking for really trying to understand what life is. And again, synthetic cells can help there. What the limits are for life on Earth, again, synthetic cells, I believe, can help here. Looking for exoplanets, synthetic cells cannot do everything. They do not replace telescopes. But they can help then bridge that gap between what we find out about extrasolar planets, as well as planets and moons in our own solar system, and what we know about life on Earth. And so I believe that that's another place where synthetic cells are really going to help to revolutionize NASA's work. And finally, once again, the evolution, the fact that we cannot rerun the clock, but we can rerun it in our labs, and this should help us find life elsewhere. And we were told to close with an acknowledgement, and there's so many people who have helped me, and our funding sources, and being a civil servant at NASA, it's all been NASA or through the TRISH, Translational Research Institute, which is ultimately NASA funding as well. So again, thank you very much for the privilege of speaking to your group.
Thank you very much for your presentation. We will move on to our next presenter, who is Dr. John Glass, the synthetic biology professor and campus director of the J. Craig Venter Institute at La Jolla. John, I think you're still on mute, actually.
John, you're still on mute.
Am I audible?
I heard something there.
Am I audible now?
There we go, yes.
Okay. Well, so I was asked to take part in this conference because of the work I've done at the J. Craig Venter Institute over the last 30 years. And let's see if I can advance a slide. That's not working. And I'm having to do this through Safari rather than Zoom because Zoom's not working for me today. Can you advance the slide, please? Anyway, in my 22 years at the JCVI, I've been part of a team that has had visionary approaches to grand scale engineering, genetic engineering programs. We've developed new technologies for rapid genome synthesis, as well as installation of synthetic genomes and chromosomes into cells. Additionally, one of Craig Venter's long-held passions has been to understand how life works. Next slide, please. So we set out. Next. We set out to construct a minimal bacterial cell that might serve as the hydrogen atom for biology. For decades, slide please, for decades, slide again, please. For decades, biologists have used genomically simple bacteria as surrogates for a minimal cell and also worked to whittle away non-essential genes from model bacteria in an effort to better understand the first principles of life. Realizing that these approaches were unsatisfactory, in 2002, our JCVI team decided to create a true minimal bacterium by building it as a synthetic cell. First, we determined what genes encode. So next slide, please. The approach that we took, shown here as to produce the images, the papers that I showed you a couple of slides ago, we had a three-part approach to building a minimal cell. So the basic approach we used is to assemble large DNA molecules from synthetic oligonucleotides. When we first started this in 2002, the largest thing that had ever been made was 32 kb, and it took that group years. When we first did this, we were purchasing high-quality oligos and assembling DNA 1 to 5 kb DNA fragments ourselves. Not practical in today's world. Now we just have them made from a DNA foundry. Next slide, please. So we build these things in progressively larger pieces and then park them, if you will, as a yeast artificial chromosome where we can get large amounts of this. Next slide, please, to move on to a step where we install those synthetic genomes into a suitable bacterial cell to produce a new cell with the genotype and phenotype of the cell that we want. Next slide, please. So genome synthesis. The first thing we do is we buy these 1 kb pieces and then we will assemble them. Next slide. We first assemble these commercially purchased 1 kb pieces, next slide, into, so we first do an in vitro step to make these commercially. Then we build, next slide, build them into E. coli. We'll take 7 of these and assemble those 7 1 kb pieces into a 7 to 10 kb fragment. All of the pieces are overlapping and we build them in E. coli. We then take 10 of those E. coli fragments, next slide, and all of these overlap, and we'll build them as yeast artificial chromosomes. So you get progressively larger pieces. We do this in 2 cycles. The first cycle, we make pieces that are 50 to 100 kb, and then later we will assemble these into a complete genome. Again, all overlapping pieces. And the complete genome comprises, in this instance, an entire bacterial genome plus about 3 kb of yeast vector sequences. Next slide, please. So genome synthesis, this is solved. But the hard part is booting up the genome. And it took us years to develop a technique we called whole genome transplantation, where we take a complete genome that has a tetracycline resistance marker. Next slide, please. And we will treat that genome. Next slide. We will treat that genome and the recipient cell, which is a species close to the phylogenetically close to the genome that we made. We treat the recipient cell with polyethylene and glycol, polyethylene glycol and calcium. This fast charges on the cell and the genome and also changes the fluidity of the membrane. In essence, it's sort of a chemical transformation. We don't really understand how it works, but somehow, next slide, the genome makes it into the cell. We then return the cell, next slide, to growth conditions, and you get a transiently diploid cell, next slide. And so this grows, divides, next slide, and we will treat the cell with tetracycline, next slide. This will kill the cell that doesn't have the synthetic genome, and you're left with a cell with the genotype and phenotype that you're looking for. Next slide, please. So we're still at the dawn of making synthetic cells. Next slide. As I said, we can synthesize genomes. We can synthesize any bacterial genome, probably up to 10 megabases, my bet is. Next slide. And the reason I say this is several years ago, a Chinese group was able to assemble 16 yeast to build a yeast strain where all 16 chromosomes had been assembled into a single 13 megabase genome. There are even methods now. So originally we did this in a single microfuge tube. Next slide. But there are approaches now, commercial organizations that are thinking about using nanofluidic technology. Avery Digital, for instance, they have nanofluidic technology, a handheld device that would take in, you would put phosphoramidites to make synthetic DNA into the device and digital information. It would first make oligos and then produce them into progressively larger and larger pieces of DNA. In theory, they would be able to produce thousands of different versions of a genome on one of these nanofluidic chips. Next slide. But the problem then would be, next slide, how do you boot those things up? So as I said, we don't understand how transplantation works. It's hard. Whole genome transplantation at present only works for mycoplasmas. Next slide. We don't know so much about how cells function. For instance, transplantation works for mycoplasmas. I don't believe we were so smart or so lucky that we picked the only cells on Earth that this works for. We think we can learn how to do this, and we're in the process of doing this now to expand this to other cell types. But there's so many things we don't know about cells. For instance, next slide. It has been an assumption in the synthetic cell community that we would be able to make a universal chassis and there would be universal parts that would be interchangeable. I think the diversity of biology makes this scenario really unlikely. Moving on a little bit, next slide. So when we first built our cell in 2010, it was called a synthetic cell. Next slide. But the definitions change. Today, this is not a synthetic cell, but rather a cell with a synthetic genome. And so next slide, what we're now trying to do, I'm doing this with Kate Adamala at the University of Minnesota, is to build a living synthetic cell from non-living parts. So the idea here is we're going to take cytoplasm from an existing cell. Next slide. We're going to add some plasmids to this that would allow this cytoplasm, a transcription translation reaction, to encode transporter plasmids. Next slide. And then we will make a microfluidic device. Next slide, where the orange part would be the cytoplasm. The cytoplasm would be encapsulated in lipid vesicles, lipids made from catalog chemicals. And so we would put these into small vesicles. Next slide. And then the process, next slide, would be to install a synthetic genome, and we'd use our genome transplantation technology. Next slide, and hopefully, next slide, we would get in a couple of days, we will get synthetic cells. These would be synthetic cells now, although years from now, we might not call these things synthetic cells because we want synthetic cells made from completely chemically synthesized parts. Next slide. So as I see
John, we're at a 1-minute warning, by the way.
We're going to be taking synthetic cells, for instance, using them as intracellular bacterial endosymbionts. For instance, next slide. We already have endosymbionts in our cells. We have mitochondria. Plants have chloroplast and mitochondria. These organelles started as intracellular bacteria and have evolved into their current forms. I'm now advising, next slide, please. I'm now advising 3 groups who are trying to use our synthetic cells, next slide, where the cells would be engineered so that they could be installed into mammalian cells and provide new functions. Next slide. So for instance, we could take our mycoplasma cells, which are not pathogenic on their own, and they might be engineered to solve a variety of metabolic problems. Still a further example of synthetic cell technology, next slide, please, is the idea of building human artificial chromosomes. And so this is a field that's been around for about 25 years, where the idea is people would take synthetic or semi-synthetic chromosomes, put them into cells, and that human cells or mammalian or plant cells, you name it, next slide, could be reprogrammed in a grand scale way, not just installing a few genes with CRISPR, but millions of base pairs of new DNA going into the cell. Next slide. So the technique has been around for a long time, but things, next slide, things we could do are, for instance, next slide, next slide, next slide. We could do a lot of things with this that would allow us to do much more aggressive, next slide, let's finish this slide up, much more aggressive things than we can do currently with CRISPR small genome adaptations. Next slide, please. And so the way this used to be done before around 2019, they would take existing small human chromosomes, whittle them, and then re-engineer them, put them back into mammalian cells with a horrible technique. Next slide. Later on in 2019, Ben Black's group and others have been able to make alphoid hacks with synthetic or semi-synthetic centromeres and put them into mammalian cells. Next slide. So our group at the JCBI, and one problem with these hacks is they multimerized in cells. You get 20 to 40 copies. Next slide. So we developed an approach where we took a synthetic hack that was more than 1 million base pairs. We put in 1 million base pairs of stuffer DNA in order to get the size up. And we developed the same approach that we used to build a bacterial cell. We use polyethylene glycol to fuse a yeast cell with a mammalian cell that installed the genome. Next slide, please. And next slide, next slide, next slide. Sorry. Next slide. So we went through this elaborate process. We were able to make hacks. And one of the great things about this, because of the size that we added, this is now a single copy hack inside the mammalian cells, which I think may rejuvenate this field because you can control gene expression copy number and produce the things you want. Next slide. So I would be remiss, given how much discussion there has been lately about this topic of mirror bacteria and potential catastrophic risk to the world, if people were to someday in the next 10 to 30 years make synthetic cells that would be capable of living, to make synthetic cells of reverse chirality. D amino acid proteins, L nucleic acids. Next slide. I was part of a group of 38 authors that published a paper in December where we presented the risk of mirror life. The concerns were that we realized, because of collaboration of synthetic cell biologists along with immunologists, ecologists, a variety of other scientists, that mirror cells that would be capable of surviving on, say, salts and glycerol, and there are E. coli strains now that can live comfortably on salts and glycerol, that such cells could pose a catastrophic risk to the world. They would be able to evade our immune system, evade proteases, and could end up, in essence, killing all life on Earth that had an immune system and causing catastrophic changes to our biosphere. The scientists that were made aware of this risk, we all came to the conclusion that it is in our best interest, the interest of the world, that we collectively decide not to do this, to keep this possibility always 10 to 30 years away. And I think we, as the synthetic cell community, as scientists, we should applaud ourselves because we have recognized that just because something could be done, it doesn't have to be done. And we have said that this is something that we don't want to do, and we want to make sure that enabling technologies are not reached that would get us to the stage that we can, that someone in the next 10 to 30 years might be able to produce such a world-ending sort of organism with such catastrophic consequences. With that,
John, in the interest of time, we will need to move on to our next speaker. Sorry.
Here. Next slide, please. So it does take a village to make a cell. A lot of folks. Next slide, last slide. And I've had funding from a lot of different organizations. With that, Cameron, I'll stop. I hope I didn't go too far over, and it's all yours.
Thank you very much, John. We are moving on to our next, our third speaker, Dr. George Church, who is the Robert Winthrop Professor of Genetics at Harvard University. George, we also still have you on mute.
Here, you can hear and see now?
Yes.
Okay. So I am going to talk about semi-synthetic cells, since I don't think we're that close to making cells from atoms. These are some of the acknowledgments. I'll have many more along the way. We set out to and I'm emphasize applications of these things, because applications both drive how fast this goes and why we're doing it. So 3 of the things we set out to do back around year 2000 was biocontainment, virus resistance, and non-standard amino acids. And 2 of the companies that spun off from this, Grow and Pearl, included some of these things. We did this by, we don't really differentiate between synthesis and editing. Most synthetic genomes, even wildly different ones, only involve a small number of edit distance from known things. But we're open-minded to this. These are some of the papers that we brought out, thanks to some of these first authors here. Some of the uses of non-standard amino acids include click chemistry and other kind of covalent bonds that are orthogonal to normal biochemistry. There's photoisomerization. Here's redox amino acids that have resulted in a 10-fold improvement in redox reaction rates. We have now nucleopeptides. These are somewhere in between nucleic acids and proteins, and they allow us to do base pairing. That's working. We have fluorogenic amino acids that unlike GFP, are only fluorescent in a particular environmental context, and they're very compact. It's a single amino acid rather than a large protein domain. Again, thanks to Mikhail, Helena, and Erkin. These are, and Externa is a new company that's working on these non-standard amino acids. One of our original goals that we set very early was to use recoding of the genome so that we could get resistance to all natural viruses. We did this not with non-standard amino acids, but with flipping 2 radically different amino acids. So two serine codons out of the 6 serine codons total were made null, meaning they were not used in any protein in the whole genome. This is also thanks to Jason Chen's group, which we used this strain as a starting point. After changing it to nothing, you could then change it to leucine. This, however, required a very strong leucine promoter and tRNA, which we got from a virus. Using the E. coli leucine tRNA was not adequate. So every time it ran across these 2 previously serine codons, TCA or TCG, it would replace it with leucine, which meant that essentially every protein in every phage was broken in multiple ways with this radical change from serine to leucine. And so you can see that we, in this far right end of this bar chart, we are getting undetectable escape from this. And we think in theory and in practice, there's no naturally occurring virus nor any evolved, any pathway to evolution. We'll keep our open mind and we'll keep testing it, but we went beyond all the laboratory strains to which it's resistant and isolate a number of naturally occurring ones. Now, I'm bringing this up all in the context, the subcontext of this meeting, which is about containment and safety and so forth. This is part of safety, but we also wanted these naturally occurring, these organisms have been engineered to resist naturally occurring viruses to also be biocontained themselves. So they might have a selective advantage in the wild, and we wanted to make sure that they were on a short leash. And the best one we've come up with so far, we've tried many of them, this is again thanks to Akos, Svena, and Reagan, was using non-standard amino acids. So the 2 standard amino acids, we did the swap, but in addition, there's a non-standard. So a total of 3 codons were used for these, for both the swap that made them virus resistant and the BIP-A, which is a biphenyl, a very bulky amino acid, is incorporated efficiently and is very inexpensive, and we evolved it so it could use very small amounts of it. Anyway, we and Fairnice's group in 2015 established undetectable escape rate, less than 1 per trillion in both laboratories, using different sets of goals, different sets of genes. But the key idea is that essential genes could be made dependent upon non-standard amino acids so that they wouldn't grow without them. And below here are a list of some of those genes and combinations like ADK, adenylate kinase, phosphoglycerate kinase, tyrosine aminoacyltyrosine synthetase. And in fact, the synthetase that incorporated the non-standard amino acid, BIP-A aminoacyl synthetase, these were all made dependent on BIP-A. And when we get out to 2 genes, we had at least 1 combination of strain which was undetectable, and then 3 genes, roughly 5 orders of magnitude lower escape frequency. So this could easily be 10 to the minus 16th. We could only measure it to 10 to the 12th so far. And then this is updated in a bioarchive paper from Anush is the first author, showing more sophisticated aspects of this biocontainment. Now, what might motivate the field, and one of the reasons that we think that engineering cellular systems, however radically, I mean, we could even get them to new solvents, radically new amino acids and so forth. If we start from a living cell, we can change it to almost everything. That's been our experience so far. Obviously, we haven't gone that far. But anyway, one of the motivations is non-ribosomal peptide synthesis is the source of a lot of our more amazing pharmaceuticals, and they have the disadvantage that each amino acid you want to put in. So, for example, here's an example of a peptide that's been circularized naturally, where 7 of the 11 amino acids in it are D amino acids, and so this is. You require all these non-standard amino acids, including D amino acids, in order for it to do its function, whether it's signaling or antimicrobial or what have you. And so, in order to get these non-standard amino acids, we essentially need at least one free codon, sometimes 2, per amino acid to get them incorporated, like the virus-resistant biocontained strain I just talked about. And so that in 2013, we came up with the first strain that had 1 free codon genome-wide. First, we make it null, and then we make it to a new non-standard amino acid. And then in 2023 and 2025, it was 2 and 3, and the 3 was the one I just described, virus resistance. There's a couple now that are getting into 7 free codons, and we have a paper we'll be submitting soon where we think we have a system that will do 34. It's not fully in vivo yet, it's mostly in vitro, but it looks like it's a good pathway, and I'll show you how that works in just a second here. So this is, we feel that we've worked on in vitro ribosome synthesis to try to get to a break-even point where 1 ribosome can make at least 1 or 2 ribosomes. This is hard, I think, because of the concentration and the structure of the cytoplasm. As soon as you break it, the cell starts going haywire. And if you try to start from something more synthetic, it's even worse. But I'm not saying anything's impossible. I'm just saying we've worked on it for many years, and it's hard. So, and these are 2 of our most recent papers on making pure translation systems. But what we've now done is we've made a cell within a cell. We have 2 systems running simultaneously. One of them is the ordinary ribosomes and their synthetases and tRNAs that are doing the heavy lifting of making proteins of all sorts that keep the whole cell running. And then an orthogonal ribosome that isn't burdened with keeping the whole cell running, but it's taking advantage of the incredible structure and density of the cytoplasm to make functional ribosomes. And here we have orthogonal messenger RNAs that are recognized by orthogonal small ribosome subunits that then allow it, and then the two subunits are covalently connected thanks to work from Mike Jewett's lab. And then we can have these orthogonal tRNAs that have not the standard CCA 3' end, but a CGA. And all this works together. And I just put in these two references to remind us that moving into a realm where we could have all the D amino acids in one system is something that has to be taken very seriously. This is thanks to Felix and Hannah. And here we thought that one of the major, so all this is working in vivo and in vitro. And one of the things we were worried about was that changing the CCA to CGA to make this orthogonal tRNAs would be challenging because CCA is the most conserved sequence in the biosphere. It's present in every tRNA and every organism, as far as I know. So we explored whether we could get aminos. We thought we might have to do a lot of engineering, which we're getting quite good at as a field using AI. But nevertheless, we wanted to see whether it was easier than that. And we did all possible variations on the CCAN in lots of different species of organisms and different amino acids. And this heat map, I'll just quickly summarize, is we can get just off the shelf without any protein design alternatives to CCA. And we focused on CGA for essentially every amino acid. That will allow us to make D versions of every amino acid. So just I want to finish on saying that semi-synthetic cells are likely to happen much faster, both because of the economic motivations, some of the applications I've described, that there's a lot more experience with it, and this problem of the highly densely packed and highly structured cytoplasm is hard to recreate. We're going to keep trying, but in the meantime, we think we can make semi-synthetic cells that can go almost anywhere in various steps. So we published recently this way of doing bioproduction, biocontainment, resistance in a real working semi-synthetic cell. We think that mirror proteins have been predicted by some of the teammates here in the paper that John just mentioned and in this meeting to be 10 to 30 years off. I think that using some of the methods I just described, it might be 5 years off in the process of trying to make ribosomal replacements for non-ribosomal peptide synthesis. So I think we've seen that with synthetic DNA in 2004, these references down below, gain-of-function virology, germline editing, in every case, moratoria were on top of all the existing rules, like the FDA rules, and they were ignored. So I think we should be skeptical. We should still try for consensus and moratorium, but I think in addition, we should have surveillance of all DNA synthesis requests, and this has been well implemented in IGSC and securedna.org for checking. A new thing is checking that rather than trying to figure out what the order does, having the person, the scientist making the order, state what they intend to do and how, and then checking that the actual DNA is fully aligned with that statement and licensing. Then we record each transaction for checking for all favorable, unfavorable later outcome. This is something that David Baker and I just proposed, and doing that in an encrypted way so that everything is secure until something bad happens. We need to have penalties. These have to be stiff and globally enforced. It's not just sufficient that we have some kind of moratorium. We need to encourage whistleblowers, which could have kicked in for some of these things that happened in 2011, 2017. And we need frequent tests with red teams to test numbers 1 to 5. Now, this has happened a couple of times, but not in any systematic way. So that's, hopefully I'm a little ahead of time. Thank you. I'll stop sharing.
Thank you very much, George. We appreciate it. Our final speaker for this first session is Dr. Elizabeth Strahalski, the leader of the Cellular Engineering Group at the National Institute of Standards and Technology.
Good afternoon, and thank you so much for the opportunity to be here. So I founded and lead the Cell Engineering Group at NIST, which is our National Metrological Institute for the US. So this means that we're tasked with developing measurements for engineering biology, including synthetic cells, within the broader NIST mission of advancing measurement science, standards, and technology to enhance our economic competitiveness and, importantly, improve our quality of life. So my group aims to use biology to measure biology, and synthetic cells, I think, will likely form the basis for some of the living measurement systems we hope to develop to help us measure and control life. Uh-oh. Not sure if it's advancing. Okay. All right. So we have projects in a number of areas, but while we've been anticipating the impact of synthetic cells and related enabling technologies for some time, I want to be clear that we unfortunately have not yet had resources or funding for investing in this area. So as a consequence, our program at NIST in synthetic cells has been limited. And personally, I find it impossible to imagine a future for biotechnology without synthetic cells. And so my hope is that we can grow our efforts. But the measurement challenges here are daunting when we consider all of the different forms and functions synthetic cells might take. We often conceive of chemistry and biology as neatly separated domains, but the closer we look at the space in between, the more possibilities we see for engineered living systems. Understanding the spectrum out in nature is vital, at least insofar as we want to use extant life on Earth as inspiration, because it can teach us how we might go about building synthetic cells, what they might look like, what they might do for us. We might also see synthetic cells that make fuller use of the periodic table than extant life, or that incorporate fluidics or nanotechnology or other abiotic interfaces. And so all of this needs to come together to inform fit-for-purpose measurements and best practices for safe, ethical development and use of synthetic cells. Now, NIST has been developing measurements for cell-free expression systems, which are expected to form the basis for synthetic cytosol and many different approaches to build synthetic cells. Reproducibility and comparability have been a work in progress for these systems, as has scale up to industrially relevant volumes. These measurement challenges, I think, will largely carry over to building synthetic cells, so it's important to address these. And with regard to measurement assurance for the components of synthetic cells and the cells themselves, I think we also need to be realistic about metrics and specifications that take into account their dependence on relationships between their parts and subsystems in context, in their environments. This might be somewhat less reductive than we might like. You know, so much of synthetic biology continues to rely on empirical iteration, which is not surprising when you think about it, because this is what nature seems to do. It's arguably how we got here, but we don't have billions of years to evolve the solutions that we need today. So we'll need to apply our rapidly emerging capabilities in lab automation and AI to tease out rules, insofar as we can find them, for practical, generalizable, predictive design and for forward engineering. And we'll need to do this in community. I think this is so important, on open platforms, to share protocols, to share materials, to test each other's methods so that we know what works and what doesn't. And all of that information and data is readily available. We've already heard quite a lot on applications of synthetic cells. I just want to add that cells as a manufacturing platform has long been a vision of synthetic biology. And I expect that our work on cell-free expression systems can support maybe finally realizing this vision in a new way when coupled to synthetic cells, to bring about a new way of biomanufacturing. Just like yogurt makes more yogurt, we might also consider using synthetic cells to enable a kind of distributed manufacturing where, say, your shampoo makes more shampoo for you in your shower while you're at work. While this and other visions for applications of synthetic cells are compelling, we of course have to think about safety, surveillance, and containment. I agree with quite a lot of what George just said. In general, we've learned that everything we make as humans tends to end up in the environment and maybe even in us to some extent over time, right? Everything goes somewhere. So we'll need trustworthy quantitative measurements to support informed decision-making, risk frameworks, and safety studies. And I think we need to do this with the sober acknowledgement that strict biocontainment, George's excellent work from his lab notwithstanding, is likely not attainable. Nothing is without risk. So we have to decide what risks, grounded in measurements, would make building synthetic cells worthwhile. There's a lot of value to be had here. How would the existence of synthetic cells satisfy our drive for discovery, or help us solve pressing problems in the world that we have, or tell richer stories to each other about what it means to be human? Or are we ready to invite the risks of not building synthetic cells? Because we could choose that too. But then we would be ceding our control and responsibility by allowing someone else to build our future for us, because someone is going to build these. And then we would have to live with their decisions about which synthetic cells to build, which applications to pursue, and who gets access to that technology. So regardless of who stakes a claim to the first synthetic cells, NIST can still contribute tools and methods for measuring them. And we're here and hopefully ready for that if and when we are resourced to do so. So thanks so much for your attention. I would like to warmly acknowledge the Cellular Engineering Group and my other colleagues at NIST, as well as our many collaborators in scientific research and metrology. And I will mention that most of our funding, such as it's been, has come through NIST. Thank you.
Thank you very much, Elizabeth, for your talk. And thank you for all of our speakers in this first portion of our session. We will now take a short break and we will reconvene at 1:35 p.m. Eastern Time for the second session of speakers. We are ready to continue into the second half of our session, Frontiers of Synthetic Cells, Applications, Insights, and Implications. As a reminder, the speakers will have 7 minutes to present, and we will keep to this timing. After this session, we will have a Q&A session with all of the panelists. To begin our next session, we will hear from Dr. Chi-Ming Tan, Professor of Biomedical Engineering at the University of California at Davis.
Hello, everyone. Thanks for the invitation to speak here. Next slide. All right, so there's usually two modalities or kind of synthetic system that we design to tackle chronic diseases. The first kind of pure material-based approach. And this is a kind of system like nanoparticles, hydrogels, where you encapsulate drugs in them and then deliver it to patient and to deliver the drugs. The pros of these systems are they're usually easier to manufacture. They are pretty well-controlled systems. The cons is you have to keep delivering these kind of nanoparticles or gels over time to treat disease for a long time, and they don't have sustained activity like cells in vivo. The other kind of system are cell-based therapy, where cells can potentially sustain drug delivery in vivo. But the cons is, unlike the material-based system, they usually lack stability in vivo. They usually cannot maintain function or numbers as stable as the pure material-based systems. And like many speakers have talked about, you also have to think about the risk of uncontrolled proliferation or differentiation of cells. So as a result, we need new paradigm for treating the diseases. One second, I'm trying to learn how to control the slides. Oops. Can I go back? Okay, so this is a non-scientific, my non-scientific categorization of a synthetic system that we see in the literature. On the y-axis, you have active functions. Things that have active functions like chemotaxis, protein synthesis, dynamic response to environment. On the x-axis, you have system of different kind of growth ability, cells that can replicate fully. This is your E. coli, BL21, for instance, or your HAC293 cells. These are wild-type cells that most labs are using. And then to the right extreme, you have systems that do not replicate at all, such as extracellular vesicles and some of the basic artificial cells that do not replicate. Or if you heat kill cells or you irradiate cells, then they also do not replicate. And on the right extreme, top right extreme here, you have potentially non-dividing system, but they can maintain all the active functions that synthetic biologists desire. And a lot of artificial cell systems are trying to approach this space by enabling artificial cell to divide. There's also people approaching this from the wild-type cells by differentiating cells to the terminal state and to make cells non-dividing, but have some of the desired function for therapy. Now, this space is, if we can reach this space in a synthetic manner, then I argue that we have systems that are potentially safe. They have complex functions for therapy. They are active as well, unlike dead cells, and they can potentially achieve long therapeutic durations. Now, the question is, how can we reach this space robustly and easily? Doing differentiation is challenging. The efficiency is quite low, and many labs are trying to create custom protocol to differentiate cells, but efficiency remain in the single-digit percentile. Doing artificial cell replication is also quite challenging. So my lab came out with a method, and this is a material-based method. It's the first time we put materials into cells. I call these cyborg cells, and it's achieved by intracellular hydrogylation. I tell my kids these are JLo cells. So just imagine putting JLo into cells. And if you do this in the optimal level, then we can achieve a semi-living system that non-dividing but active. And in the previous publication, we have shown that using E. coli, we can achieve bacteria that are non-dividing and but yet can maintain metabolism and protein synthesis for a few days. They also resist some of the stresses that are often encountered by bacteria in applications such as high pH, hydrogen peroxide, antimicrobial agents. And in later publication, we have extended this to different kind of pathogens like Pseudomonas, S. aureus, Klebsiella. It take a while for me for the slide to change. And in some of the work that we are wrapping up to publish, we have applied this kind of cybobacteria as cell and immune system programming tools. On the left, you see cybobacteria being used as vaccines. So pretty standard protocol of testing with mice, vaccinate mice with the cybot Pseudomonas, challenge mice with the Pseudomonas, and we look at survival and safety. All mice vaccinated with the cybot vaccines were safe. And then if you look at the red line in the survival curve, more than 85% of the mice were protected from the Pseudomonas infection. The blue line is hit-kill bacteria. The gray lines are, the dotted lines are unprotected, unvaccinated mice. And clearly, the non-dividing but active bacteria is superior than the hit-kill Pseudomonas. And we are currently moving towards S. aureus and also tuberculosis. On the right, you'll see using cybobacteria as synthetic organelle inside mammalian cells. And we are also wrapping up this work. So what we are trying to do here is learn from this idea of endosymbiosis, but do this in a synthetic manner. What we have been able to show in one of the sample results is we show that SKBR3, these are breast cancer cells, when invaded with cybobacteria, this is cybot salmonella, and most of the mammalian cell, the SKBR3 cells, were protected from this mitochondrial stress from the FCCP. And the higher the dose, the FCCP, the protection is less because now you get into the very toxic regime that the bacteria can no longer protect the cells from the stress. And we have other results here where we show that the cybobacteria as organelle can serve as biosensors and also enzyme factory inside the mammalian cells. Now, all those previous work was done with bacteria. Now, can we do this with cyborg human cells? So we have been able to convert a number of human cells into the cyborg version, non-dividing but active, that include placenta MSC from human donor, T cell, HEC293, primary GBM cells from donors, and also 41 breast cancer cells. Just a snapshot of the results, we have shown that the cyborg mammalian cells do not replicate but maintain activity. On the right here, you see secretion data where cyborg cells at the per cell level can secrete some of this molecule more than the control cells. These are cyborg primary mesenchymal, placenta mesenchymal stem cells. And we have also shown that the activity maintained for more than 5 weeks. We are doing quite a number. We have done quite a number of ASA to characterize these cells from what you can see in the cell biology literature, all the way from high resolution imaging down to genomic assay, etc. We also shown that cyborg PMSE are resistant to quite a number of stress that these cells usually encounter in applications such as RS, hypoxia, hydrogen peroxide, and staurosporine. We also shown that the cyborg PMSE are useful as therapeutics. And this is a cell culture assay where on one hand, we stress the neurons. And on the other hand, we try to rescue the neurons by supplying the stem cells and the cyborg PMSE. And you can see that the cyborg cell can rescue the neuron from the stress. And finally, we show that the cyborg cell can implant in immune competent mice for the first time using cyber PMSE. This is human PMSE cells being implanted in mice, and as expected, wild-type human cells will last in 3 weeks, but the cyber cells, amazingly, we're still trying to understand this, maintain themselves in this immune compromise for more than 3-4 weeks, while also secreting a therapeutic. Finally, thanks for the funding source and the lab members. And I also just want to quickly call out something along the line of what Joan has mentioned. We have recently published an treaty in the Spiroacioloma on synthetic human chromosomes and the consideration of ethics and global discussion needed along this research area. Thank you.
Thank you very much, Ximing. We appreciate it. Our next speaker is Dr. Michael Booth, the Associate Professor of Chemical Biology and Organic Chemistry at University College London.
Hi, everybody. Thanks for the invitation to come and talk all the way from across the Atlantic Ocean. I'll see if I can get a laser pointer working. Yes, my name's Mike Booth. I'm an associate professor at University College London, and I work on synthetic cells that are a bit different to what has everyone spoken about already. Lynn did mention the bottom-up synthetic cells, and so hopefully my slides will go forward. There we go. Okay, great. So synthetic cells can be built in all sorts of ways, which you'd heard about already. So a lot of top-down approaches you've heard about from using the nomenclature that Lynn had spoken about in terms of from living cells. But what I do is I build synthetic cells from the bottom up. So these are all from components you can get in the lab, and then just throw them together and try and make some form of cell-like system. So for me, a synthetic cell is something with a cellular function in a minimal environment. And so often what we're doing is we're making compartments with lipid bilayers, just like a cell. We're putting DNA templates on, which is like a synthetic genome, but just with maybe 1 or 2 different genes instead of the whole genome. And also including things like cell-free protein synthesis systems. So we can do transcription and translation within this minimal compartment. So this is not living. And so the advantage here over all the systems you've heard about before is it's much more customizable, right? We can include lots of biological parts, just like what you've heard about, but we can also include lots of synthetic parts. And this is what I'll focus most of my talk on today. And again, coming back to this idea of ethics and risk, one major thing about the bottom-up synthetic cells is they're not alive. I mean, there are people going to try and actually turn them alive or, get across that boundary of non-life to life. But there's an advantage also to not having them be alive, right? Because in this case, we actually don't have as many of the drawbacks and ethical problems that a lot of genetically modified organisms might have. But you still have a lot of the applications, right? So you still be able to study living processes in a minimal environment without all the crosstalk from in a cell. You can create potential smart therapeutics, biosensors, bioreactors. And I think this is an important point that I like to make, that by having these synthetic cells, you've got a niche between the idea of a living cell and individual molecules, right? You've got something new that can fit a new type of application beyond either of those. And so one thing that's absolutely vital for these sorts of systems is control. It's so important to be able to control their activity, right? Because basically in any activation you can imagine, you don't want them being active either before they've reached the target site or even for too long or in the wrong place. And so what we tend to do, one of the main things we work on is methods of control. And to do that, we do chemistry, right? So I'm in a chemistry department. And so we do a lot of chemistry. And what we tend to do is we work on chemistry of the DNA template, right? We modify the DNA template to make it controllable. And as you can imagine from synthetic cells, to be able to control them, it's actually quite difficult because you need to get across the membrane to control them. And so things like small molecules are not that easy to control mechanisms because they don't all go across the membrane. Things like pH changes, they don't go across the membrane very well. And so things that are remote control that can go across the membrane is what we focus on. And one of them is light. So light is fantastic. You get amazing spatial precision, better than any other stimulus. Biocompatible. One thing that's unique to light is multi-wavelength orthogonality, right? Because you have different colors of light that have potential to activate with. Tissue penetration is a problem with light, but once you get to longer wavelengths of light, you do get some form of tissue penetration. And so most of the systems we've been developing are on this sort of design where we've got a promoter of a gene of interest. So this is just a synthetic gene and promoter system that is just blocked with a protein and photocages. And so what we've managed to generate is different types of photocages, so different photocage chemistries that allow us to unblock that with either UV or blue light orthogonally. And so what this allows you to do is to get a pattern. So here are synthetic cells. Each of these dots is a synthetic cell that's been activated with a photomask. So you can see the spatial precision of the patterning you can do with light of activating, in this case, just fluorescent protein expression. And in this case, you can see an example of the orthogonality of light. This example, the only way of doing this with dual wavelength patterning is with dual wavelength patterning of light. So we've got a system where we're expressing two different proteins, but they only have the function when they're present together. And so we can see is that only when the light beams overlap do you get the synthetic cells that are now active, right? So you get incredible precision in their activity. So we've also managed to do activation between non-living synthetic cells and living cells, right? This is obviously what you really want to be able to do. You want to be able to control how synthetic cells interact with the environment, ideally with living systems. And so I've tried to simplify this as much as possible for everybody. So we have signal producing sender cells, and these are synthetic cells, and then signal detecting bacteria. And so here we have a DNA template that is light activated that produces a signal synthetase, right? This can make a small molecule in the synthetic cell on demand, and that will make a signal. And this signal is membrane permeable. It's just a natural signal molecule that bacteria can use. And then bacteria has a system inside it that can then detect that and express a fluorescent protein. And so what we ended up doing is putting these all into one system together. So we've got the synthetic cells and the bacteria co-incubated. And then if we don't shine light and we don't have any DNA in the system, the bacteria do not express anything. If we have the signal synthetase DNA, but we don't shine light, we don't get any activation. But only when we actually then have the DNA and express and shine a light do we then get the expression of the synthetase, which makes this signal molecule, then goes into the bacteria. You can see these bacteria are now turning green in these microcolonies because they're responding to the synthetic cells. You can see the synthetic cells are still here in the agarose layer below. Right, so this is the first example of being able to actually control remotely non-living and living synthetic cell communication. So something else we've been doing recently is going beyond light. So light, as I said, is really fantastic, but there's real problems if you want to actually then use it in vivo or anywhere where there's deep tissue, deep penetration, or somewhere you can't necessarily see, for instance, obviously. So this is where things like magnetism come in really handy. So they're deeply penetrating, biocompatible, and they're actually already, if you use them with nanoparticles, it's already been applied using alternating magnetic field. And one thing that's really amazing is that they're also trackable if you're using nanoparticles, right? So you can track them, so you can image them and potentially activate them. And so to try and explain how this works is a bit more complicated than the previous example. We have these iron oxide nanoparticles that we coat with DNA, right? So the green strand of DNA is a promoter sequence, just like in the previous example, we have a promoter. And if we put an alternating magnetic field on the system, what happens is that we generate a local temperature, very, very local to the nanoparticle, that increases the temperature just outside the nanoparticle surface and then denatures this DNA. Right? So we have a release of this promoter sequence. And then if we have a DNA template that encodes anything, and then a promoter sequence that is inactive, single-stranded, then what will happen is we can form the double-stranded template and then activate gene expression. And this is what it looks like. So this is similar to the previous example, but now this is with magnetism. So we can see the SNCC cells in purple here. And if we have all the system together, but we don't apply a magnetic field, we get no expression of the, in this case, the M-neon green fluorescent protein. But when we do apply a magnetic field, we get a lot of the cells now expressing protein. So we're getting this really tight control of cell-free expression in synthetic cells, but with a really deeply tissue-penetrating magnetic field. And just to give you another example of something we've done with that, because obviously what you'd want to do is you want to use that as a drug delivery system potentially. And so what we ended up doing is putting this whole system, but now instead of having a fluorescent protein, we're expressing a pore protein. So this DNA, all we have to do is just switch out the sequence to a different sequence that expresses a different protein. And if we put these small molecule cargo inside, so you can see the green now here is actually just a small molecule fluorophore cargo. And if we don't, if we just leave it at the beginning, we can see that there. If we don't put a magnetic field on the system, the cargo stays inside, right? As you would imagine, nothing's happening, no pores being expressed. But if you put a magnetic field on the system, you then release all of that cargo. So we've got this controlled release with a tissue-penetrating stimuli, right? Showing the potential of using these non-living synthetic cells as drug delivery devices. So that's finishing for me. I left a lot of the ethics stuff we can talk about later, but really I want to focus on the idea of these bottom-up synthetic cells and using chemistry to be able to put non-living chemical parts into these synthetic cells to give extra functionality. And thanks to my fantastic group and funders and you for your attention. And I'm happy to answer questions later. But
Thank you very much. And yes, we will have questions after the next two speakers. So please make sure that you are putting those into the Slido. Where's my notes? There we go. Our third speaker for this session is Dr. Neha Kamat, Associate Professor of Biomedical Engineering at Northwestern University.
Thank you. All right. So like Michael, thank you for the invitation to talk a little bit about synthetic cells and join this conversation. So like Michael, my interest is also in working with a very bottom-up approach to designing synthetic cells. And in my group, we see a really optimal design for a synthetic cell lying at the interface of whole cells, biological living cells, and cell-free systems. And in particular, we are not interested in meeting a definition of life. In fact, I sort of see living cells as a liability. And the reason for that is that for billions of years, living cells have evolved to survive and grow. And so what that means is they are answering to a higher program to survive that can really outcompete your desired program in a given cell. And so with synthetic cells, we can sort of overcome that capability, work with highly controlled components, and get a function that is desired. Another element that we're really interested in designing is the membrane. So there are a variety of very useful membrane proteins that act as this interface between the biosynthetic machinery inside a cell and the environment. And we are trying to learn rules to really design the interface such that we can sense a variety of molecules. So to complement some of the really neat environmental signals Michael talked about, I will talk a little bit later about some work we're doing to sense small molecules. Okay. All right. And again, we see the applications in biosensing, drug delivery, and bioreactors with the advantages of being very customizable, having fewer metabolic constraints, and ultimately being designable. So when I imagine the capabilities of a synthetic cell, what I'm really looking for is something that can sense the signal from its environment. So perhaps this green molecule over here is the environment, or maybe it's another biological cell. But our synthetic cell on the right should be able to sense that signal, should process process that signal, and then respond in some way. And critically, it should be able to do that iteratively over multiple cycles. And that's something that I think we're all as a field still working on when we think of the bottom-up design of synthetic cells. There are a variety of future applications that I'm excited about. So biosensing is one, and I'm going to go into more detail about what I envision synthetic cells could be doing in that space. Another area is material integration. And so here I envision something like a submarine that has a material coating on its surface that would allow that submarine to smell underwater. Or textile fibers in which we can embed synthetic cells to give us the capability of real-time camouflage. And so those are all functions that can come from biomachinery, but that need to be complexed with materials to really enable their use in new environments and in new ways. And then the final future application that I think a lot about is drug delivery vehicles. This is the reason I actually became interested in synthetic cells as a graduate student to begin with, because up until now, even to date, all of our drug delivery vehicles, and I'm thinking lipid nanoparticles and liposomes, or polymer nanoparticles, they've all been designed to do just one really simple thing, which is fall apart. And we haven't really moved past that because we haven't really imagined or thought about how to make drug delivery vehicles that, instead of just falling apart, can move around the body, sensing and responding in really controlled ways. And so an example of how biological cells are able to do this is CAR T cell therapy, but that's expensive. And so it'd be great if we could bring those sorts of capabilities to synthetic cells. There are a variety of limitations I see keeping us from achieving some of these future visions. One right now is a limited lifetime of activity. So when you use a bottom-up approach to build a synthetic cell, you need to, at some point, reload your cell with nutrients and remove waste. And the reloading process is not trivial. There are ways that we're thinking about doing it, but it's one we'll have to overcome. Another one is the time scale. So biological synthesis takes anywhere from minutes to hours. So how can we speed up the responses so that we might get something in seconds if we want a real-time response? Finally, and I think this is the ultimate one, we don't yet understand the design rules. So what compositions of the starting components do we need to yield and give us a specific function in a specific environment? And how does a population behavior change when we have artificial cells of mixed functions put together? Those are the sorts of things we need to understand in the next, I would say, decade. Okay, so the primary advantage of synthetic cells is really the ability to design and program their behavior. Because we're starting with components that we understand completely, so cell-free systems, for example, alongside components that are going to make up our membranes, we can put these together in precise combinations, precise compositions, and start to initiate these design, test, build, test, learn cycles where we see what kinds of functions we get out of our synthetic cells and then iterate to continue this process until we really understand which starting components give us a desired function. Ultimately, we will need to use AI methods, and I believe alongside physics and theory-based computational design. The other thing we can start to do is use the same design, test, build cycle, but now design our artificial cell alongside materials. So we can co-design these two things together, and I could see the components that lead to a desired function being different for a synthetic cell when we want that function to exist in a given material. And so we'll have to do the same thing, but now within the material of interest. Okay, so I just wanted to end with a quick biosensing example. So a long-term vision I see where we could use synthetic cells for biosensing is to give us information about environments and within those environments. So right now, when we want to sense something or detect molecules in a given blood sample or a urine sample, we'll draw that sample at a given point. We can take a water source at a certain site along a stream or take a blood draw, and we'll test what is in that sample. But we're missing a lot of critical information. So a low pH near a tumor site is not something you're going to detect when you take a blood draw down, in your arm. Lead that's building up on the bottom of a groundwater stream is not potentially something that's going to show up if you are collecting a sample downstream. And so I'm really inspired by the idea of thinking about sensors that move through complex environments. They're designed to sense signals of interest, and then they're designed to transduce that signal into a recorded memory, such that if we can figure out how to collect these sensors downstream or from the body, we can look at them and we can see what they saw along the way and really get a better idea of an environment and in a whole new way. And so towards that vision, we've been thinking about how to transduce signals across membranes while keeping that membrane intact. And transmembrane proteins are a phenomenal way to try to address this. So here we started working with a nitrate sensing 2-component sensor. The goal here is really to move information across the membrane without moving molecules. And that is really, I think, the power of biological membranes and membrane proteins to date. And so what this system does is the blue protein will sense nitrate. It, in response, phosphorylates a purple transcription protein shown here, and then that transcription protein, which once phosphorylated, will dimerize and sit down on a gene to initiate gene transcription and translation. And so we recently reconstituted the system using cell-free expression components and synthetic membranes. And what we found that I think is useful to mention here is that we could tune the performance of our sensor by changing the composition of lipids around the protein. And so what I'm showing you here is a luminescent signal. So we're looking at our reporter protein that is producing a luciferase signal in response to nitrate addition. And we compared that to when we added water, which is something that the sensor should not respond to. But you can see as we change the composition of our membranes, we change the performance of our sensor dramatically. And so these in the starting materials and the starting lipids had a dramatic effect on the the function. And this is the kind of information that I would like us to start collecting so that we can design function more precisely. Okay, so ultimately what we did is a principal component analysis for all these different lipids and the resulting performance of our sensor. And a really interesting property came out, which is the critical packing parameter of lipids. And what we found is that, and this is a neat property that basically describes the shape of lipids, but we found that the fold change, the increase in luminescence in response to nitrate over a nonspecific molecule like water, increased when we added more conical lipids to our membranes. And so we're starting to understand here a design rule for this particular protein, that it likes conical lipids, and that helps its performance. And so that's something I see us doing going forward. Let's think about building understanding between the components that we start with and the resulting function, such that we can down the line think about functions that we want and then have formulas that tell us which synthetic cells to build and which components to build those with. As a teaser into our discussion that will come online later, I wanted to say that I think we should be aware that as we start to adopt AI techniques to uncover new relationships for synthetic cell function, that we aren't just generating data and building relationships without an underlying appreciation of the theory that is really driving those relationships, because that's ultimately what's going to allow us to extrapolate what we learn to new functions that exist in data sets that we don't even have yet. So with that, I will thank my group, collaborators in our Center of Synthetic Biology, as well as many funding agencies. So thanks.
Thank you very much, Neha. And our final speaker for this session is Dr. Juan Perez Mercader, Senior Research Fellow in the Earth and Planetary Sciences program at Harvard University.
I hope you can hear me, and I
Yes,
hope
we
I
can.
am. Okay, that's quite an advance. And I am in a remote location in Colorado. So That's so. My talk today is trying to explain to you or to describe to you how why and how we are motivated in building systems which behave like life, but they are not at all related to life at the molecular level. In other words, they do not use any biochemistry. But before I go on, I would like to thank the organizers for having me here today, and just let you know that I am having a great time listening to everybody's talks. So the next transparency, I'll try and see if I can do it myself. Okay, we do know, and we have known for the last 40 years, essentially, that the interstellar medium from which the solar system formed is full of chemicals, complex carbon chemicals, which probably at some point in their in some point in their history, as they collapsed in the center, okay, as they collapse in the center of some star, they began to form planets, and these planets went through a process in which the original materials, they actually collided, and they actually coagulated into some protoplanetary system. And today we know about 5,000 of those systems, extrasolar planets, and we ask ourselves, how did life start in our planet? We do know that from 3.6 billion years ago until today, Luca, the last universal common ancestor, was already based in biochemistry and in all the good things that biochemistry brings with it. We do know also that there was a stable hydrosphere in the planet by study of silicons and so on, a stable hydrosphere in the planet already 4.2 billion years ago. There is a lot of speculation how we went from a stable hydrosphere to the last universal common ancestor. In the middle of that, you have attempts to explain things in terms of the RNA world, the pre-RNA world. And then there is an era, which is the so-called protocell era, which is probably something that may have happened here and in many other places in the universe, where protocells were formed and then evolution in some way took place, and eventually that kind of prebiotic chemistry led to the pre-RNA world. There is a lot of speculation on this, and there are no really good models for this, and we have attempted to try and understand what might have happened in the very early stages, and here is what we did. We started thinking about what characterizes life, and what we want to do is see if we can actually produce chemicals or produce some system which actually has all the properties that characterize living systems. They are, as it is known by everybody here, information, metabolism, self-reproduction, and evolution, both adaptive and non-adaptive. And this has been on planet Earth since at least Luca, last universal common ancestor, and there were all these four properties. This is an important point. All these four properties: the handling of information, metabolic activity, and self-reproduction, as well as evolution. All of those are unified in the cell division cycle that we all know via biochemistry, and uses standard chemical processes. So knowing this, we began to ask a couple of questions, and the questions are at the end where you see the bullets here. So what is the simplest, or what would be the simplest chemistry-based life mimic system that you can synthesize in a one-pot reaction using small molecules? Why do we ask this? Because probably things started from a simple beginning, and they went on to a much more complicated through some evolution into much more complex systems like the ones we see here today. The second question that we set is, can we engineer, integrate, unify these all functional properties into an aqueous artificial system without using biochemistry? And then all of this sounds like a very nice set of purposes, but where do we start? So in order to start this, we went to what physics can do for you. Remember, I am a physicist, and therefore I am to be blamed. Okay, but this is the way I think. So we built a set of equations that actually are able to unify the four properties you saw there. If you're interested in this, you can look at the references indicated down there. And what happens is that and I hope that this works. What happens is that when you put these equations into a computer and solve the equations, then if you represent with the red the food, and you represent with the blue something like a cytoplasm or something like that, where the autocatalytic activities and all that goes on, then oops, it didn't work. It doesn't work. I'm trying to reproduce my transparencies. My movies, this is really bad. So the movies are not coming, that movie is not coming out. I have a few of the movies. Well, I'm sorry, this is not. So what happens is that you see that the solutions to this set of equations grow, and then they replicate, and then they die, and then new ones emerge, and so on and so forth. So it actually shows all the properties of living systems, natural living systems. So then you begin to say, how do you translate these equations into chemistry that can boot up and operate a non-biochemistry operated artificial living system? So the first thing you need to do is to actually generate a compartment so that the reactions can go on. And then we want to remind everybody at the door in my lab that no biochemicals, biological materials allowed, except the ones in the researchers. So this is just a little joke, okay? And I get very nervous when I talk via Zoom. It never works the way I want it, okay? So I'm trying to catch up. So to the rescue for making these compartments for make the life mimics comes something which was invented at the beginning of the 2010s, late 2000s, which is called polymerization-induced self-assembly, where you have a molecule which is hydrophilic, you have some monomers in solution, which are soluble in the solvent, say water, and there is some active molecule here, which is called chain transfer agent. And when you shine light on the system, what happens is that there is a polymerization that starts, and you make an amphiphile. This is a really neat trick. And this is a really neat trick that takes you into some things that we have seen in Neha's presentation. It takes you into thinking, how can you use this to actually make a system which behaves like something that starts, and then it becomes, it evolves, and then it becomes doing the things that a living system does. So we use polymerization-induced self-assembly is a way of implementing the changes in the packing parameter. Thank you for the 1 minute. For the packing parameter. And so the packing parameter is changing as the polymerization reaction takes place. And the chemistry, the systems that you need to do this are very simple and cheap. You produce systems, I am sure that none of these movies are showing, which is a bummer, but you can see them in my website if you want to. The systems self-reproduce, the systems move, they actually experience phototaxis, chemical phototaxis. They actually show a realization of the cell division cycle. Their self-reproduction is clearly non-linear. And there are many examples of such systems. There are probably, we estimate now that there are thousands of such systems, different chemicals leading to such situations. Recently, we have shown how these systems self-reproduce. You can see self-reproducing. You see the time, how time evolves. You can see how there are the self-reproduction leads to various generations of the system. And this happens in 12 hours. And these systems produce some form of cell division cycle, which it can be understood very well with the nano physics going on. Here is I'm sure, oh, this one is working. I can't believe it. So you start with you start with the hydrophilic end with the molecule. These are the monomers, and then you shine light. Then the monomers are integrated. Then the monomers, the system keeps growing with the action of light. Then you get first micelles. The micelles begin to have pores, and then they start drinking water. Inverted micelles happen inside. The system keeps growing because of the chemistry which is in there. It's a heterotrophic system. The system grows. The system then becomes unstable in the membrane, and then it spits out stuff which again is used after the cell division takes place, or the self-reproduction takes place. They do that. We can also show that they compete, and they compete by competitive exclusion, which is quite interesting. In fact, similar to what happens with paramecia. And we also showed that these systems adapt. And our view of the systems is that this is like the Chemex system for making coffee, and we want to make it into something much more complicated, like what you see here in the cover of the New Yorker for one of those automatic coffee making machines. And to conclude very quickly, the origin of life may be a virtually automatic consequence of carbon chemistry and the physics of self-organizing systems, given favorable environments. There are literally thousands of chemicals. They can exist in a large class of organic solvents, not only water, alcohol, DMSO, etc. They mostly take advantage of chemiosmosis, the hydrophobic effect, and the presence of defects on the surface. They can be adaptive and evolve by degradation, competition, and co-option of materials in their medium. There is a growing community. Unfortunately, very few people in the USA are doing this. There is a very nice and active community in China, a very active community also in Europe, and an active community in Australia. And there are applications to drug delivery, mining, microplastic management, etc., origins of life study, and so on. These are the people who have collaborated in this part of my project, projects in the last 10 years or so. And here are the founders. This one, unfortunately, is gone. And but here we are. And I thank you very much for your patience with me, and for making room for this into your community of artificial life. Thank you very much.
Thank you very much, Juan. We greatly appreciate your talk and all of our speakers for today. I will now make sure that all of our speakers are available for questions, at least looking across my screen. I see most of you all here. So just as a quick note, we will also be having questions from the audience in the Slido, as well as the committee members may also have certain questions that we can field as we go through the process. We'll go until about 3:15, so about 50 minutes of conversation. And my hope is that we'll be able to kind of hear from everybody throughout the questioning. But if there are any particular points that you'd like to make, please don't be afraid to react or raise a hand or anything like that, so that we can keep the conversation somewhat organized with what we got. So the first question that I'd like to begin with, and I think what I've really appreciated about this session has been, looking at the advances of the technology, what we're able to do with artificial cells, with these different types of chemistries, what can we learn, what can we do? But one of the questions that I think is important for this committee, as well as for the broader community, is to understand what you and your research groups are thinking about in terms of the key biosafety, biosecurity, and environmental concerns in your space. So when you're thinking about future deployment or even current development, what do you view are some of the key concerns that your group and your community needs to leverage with these synthetic cells? And I'd like to start with Lynn. And Lynn also, of course, with the large paper, I'm sure has had conversations with the broader community on this question as well.
Right. Thank you very much. This is going to be a very different answer than everyone else anyway, because most of my concern is building products for off-planet. And so unlike everyone else, we need to deal with the Outer Space Treaty. And so there are very specific requirements for us in terms of contaminating different planetary bodies. So for the moon, the assumption is it's dead, always has been, and so there's not a concern. And because the radiation flux is so high on certainly the moon, Mars, plus you've got the temperature swings and everything else, don't bother trying to go to the surface of Venus, Mercury. These are all nasty places where organics fall apart. And so it's not considered a big concern at all to have things out on the surface of the moon, because the assumption is they will be very quickly sterilized. Mars is different because of the possibility that there is even now extant life. And so there are very specific planetary protection protocols that we need to follow. And that would be true whether there's synthetic cells or naturally evolved cells. Of course, once human life is on the line, the assumption is going to be that it's human life that takes precedence. Whenever I'm working in the lab, though, it's the usual terrestrial-based stuff, and that's the sort of thing that my colleagues have thought much more about, and I'll bump it to them. But I did want to get in that we're dealing with something very different when we're off planet.
And I'd like to invite if anybody else would also like to share. I think as well, we are looking at a lot of agricultural applications, environmental applications, medical applications that we saw a wide range of that today. I'd like to hear those different perspectives as well, especially, even for, say, Qiming, you've already done animal studies with some of your cyborg cells. That raises some interesting questions about containment, since it's no longer just in your lab. It now moves to an animal facility.
I would say in terms of cell therapy, if you talk about putting synthetic cell into animal, the control proliferation is still usually an afterthought and kind of a secondary priority in most of the literature. So most of the literature still focus on demonstrating functions, especially novel ones. And then oftentimes they'll throw in afterward and say we implement a kill switch, but that kill switch to me is probably less characterized like the bacterial system that have been studied quite a lot. So that's what I view as the current state in terms of implanting replicating cell into human systems. But that's why we are working on this hybrid cells thing, where replication is control is kind of built into the platform, and at the same time, we also enhance the robustness of the system.
I will add in that the work we do is mostly with living cells, and our minimal bacterial cell, I worry little about its escape from a laboratory environment or it causing any mayhem, because as a result of its massive gene minimization, we have made the wimpiest cell on Earth. And so while mycoplasmas, the organisms on which our minimal cell is based, readily contaminate mammalian cells, culture mammalian cells, or other species, are problems in laboratory animal colonies. Our cell is incapable of infecting anything, mammalian cells, anything else. And we are now considering using this to make it a cell-based therapeutic. So in essence, what you have is a cell that you can culture and make large amounts of in the lab, and then potentially use as a therapeutic, where you would inject into a patient or an animal so that it could deliver proteins for cancer therapeutics and not survive. Also, as a result of minimization, we eliminated all of the antibiotic efflux pumps, so it becomes an order of magnitude more sensitive to antibiotics than the wild type organism. But other cells we work with, we worry about more. And it's the sort of thing that we think about kill switches. We have a group at the JCBI who's developing new kill switches. Or when we think about building human artificial chromosomes for potential use as therapeutics, the huge size of those chromosomes make it possible to think about including kill switches in those cells, whereas with CAR T therapy, such inclusion of new genes really wouldn't be practical.
I think
Is
it's
there any-
difficult to know how concerned to be about accidental release or escape of these technologies from a controlled environment or a laboratory environment, because for the most part, we aren't surveilling our laboratories or our offices for the DNA we're using in the laboratory or the organisms we're using. And so I think we need robust, validated measurement methods for understanding the baseline of our microenvironment, so that if and when there is a release, we'll know how to detect it. We can respond effectively and rapidly, and act accordingly. But up until there's data, it's hard to know how concerned to be.
Well, and something that oh, Neha, go ahead.
Oh, yeah. Yes. Lynn, can you turn it? Sorry, we're sitting together. So I just wanted to second what Elizabeth said. I with measurements and being taking surveillance. So I recently learned from a biorisk analyst who's at the conference that Lynn and I are at about the term deny by deterrence, which is to prevent bad actors from doing what they're going to do, basically by being on the defensive. And that involves taking frequent measurements, having conversations about what biothreats could look like, monitoring our water quality, monitoring our crops, and starting to think about what we should be looking for. And I think if we are having those conversations and we're learning how to be vigilant, that will actually go a long way towards preventing threats.
And to be clear, I think we're starting to have these conversations, but we're not yet doing the work needed for this, largely because we aren't resourced for it.
Yeah, Juan, please.
Yeah, of course, the systems that we make are completely heterotrophic. In other words, they need to have the actual components in the solution where they are. But after attending a Silomar this last February, I began to think about what a bad actor would want to do with systems like this. And I am concerned that it is very easy to make such systems. I mean, if we can make it in my lab, I'm a physicist. If we can make it in my lab, being a physicist, anybody can do it. And if anybody can do it, and you have a system that self-replicates or self-reproduces, as we call it, and it does it with spores that it sends into the system, it's very easy to think of situations where you come to a bad actor trying to poison a city, for example, by generating, well, I will not say how I envision that, but it can be thought of.
Yeah.
Okay, and this is very, to me, is very concerning that such systems can be built. What we have in the lab now, it will not go anywhere, okay, because it doesn't have the ability to do anything like that. But and the intention was to prove that there could be life in other places, which is not like life on Earth. But if you are a bad actor, you can get the means.
Lynn, go ahead.
I'm sorry, I just wanted to add something that we always have to be careful, though, that we're not overdoing it so that we have completely stifled research. If you look at an STS form for sucrose or glucose, it can blow up. Yes, if you have a compressed silo of it. I don't think any of us are at all concerned about having a lump of table sugar that it's going to blow up. And so we do have to be very careful. Right now, the synthetic cell systems are very much on life support. The work that's been shown today is brilliant. I've so much admiration for all the work that was shown by our colleagues, but I don't think any of them are ready to take over the world, or their cells are certainly not ready to. And so we do have to have that fine line between what is remotely possible and what is more likely, and we want to prevent against that versus completely bringing ourselves to a standstill in our research.
Michael, do you want to go ahead? Yeah.
Yeah, I was going to reiterate a point I made in my talk in terms of the difference with bottom-up synthetic cells. right? So in that case, like the release problem is not really a problem, right? Because they're not dividing, they're not living. The alternate thing though is that they could potentially be made a lot easier, right? You could just buy the components to make them, but then it comes down to the situation that already in the case, which is like screening DNA sequences, right? So that's and those things are already in place or being built better, ideally.
John, do you have another? I have a fault. Yep, go ahead.
Yeah,
Yep.
it is from the beginning of our field. So 25 years ago, when I was going to meetings about synthetic biology, about engineering cells, there was more discussion of the risk of synthetic biology than there was of actually presenting science. It's been part of our culture, and it's always been part of our culture. One of the things that we have learned from both accidental and intentional releases of genetically engineered bacteria is that the act of engineering organisms makes them weaker. They don't fare well in the environment. They just don't do well. If you want to cause, if mayhem is your goal, don't try to do it with bacteria. Make viruses. It's a lot easier. It's a lot cheaper. You'll cause more trouble. But bacteria, just by having a cell express green fluorescent protein, you slow the growth of the cell because it's putting all of this energy into it expressing a gene that does it no good. And out in the world, this is going to result in the eventual elimination of that cell. So that needs to be, I think, a thing that we keep in mind when we are talking to the world about what we are building, that it is really, really hard to make an organism that didn't evolve naturally, which will persist for very long should it escape intentionally or accidentally.
So well, along those lines, I've got a couple of questions from the committee that are coming in that sort of get into this question. We've heard again, Michael, to your point, this bottom-up approach, that risk of escape, that containment is maybe not our question, but one of the comments that came in sort of looks at how do we, escape and downstream effects really, cannot and should not be separated, right? So, even though we're trying to figure out maybe some of this containment, we really still need to think about some of those potential unintended consequences, even if that's not sort of the immediacy. But a question that came in that I think is also related to that is, what type of risk assessment questions or risk assessment framework do you leverage in your own research? And maybe the follow-on question there is, if you can't think of a particular risk assessment framework, what maybe could be in place that would be enabling to the research, I think, as Lynn has mentioned, but still allows for the protection of, our, society. Sorry to toss a hard question in there.
You have to think about what you're planning to do, and you have to talk about it with your colleagues. So in 2019, we gathered 80 synthetic cell folks at an NSF Ideas Workshop at a conference center in Virginia. And collectively, we came up with great new ideas for synthetic cell research. One of them was the idea of building mirror cells, starting efforts to build mirror life. And everybody in the room, no one objected to this. It seemed like really interesting. It would teach us amazing new things. Had there been immunologists in the room, they would have said, whoa, don't think about this before you do it. We have ideas about how this could end life on the planet as we know it. And so it is important that everything that you think about doing in the lab, really consider it carefully and don't isolate yourself. Think about the risk and ask other people to think about it if you have any concerns at all.
Neha, go ahead, please.
I might answer this by I started thinking about radioactivity and how we try to prevent radioactive, I guess, contamination. And so typically, if you do work with radioactivity, you have in place regular checks to swab random areas around your space, your shoes, and you do tests to make sure that the radioactive sample you're working with hasn't left where it was supposed to. And so maybe we think of our research that way, where we introduce barcodes, DNA barcodes into our work such that if it appears somewhere else, we now know we have a containment issue.
Towards that end, when we built our first cell with a synthetic genome, we developed a 4-base alphabet, and we signed our work. We put watermarks in the cell so that we could always distinguish it from what might be a very similar natural cell.
Ming, I think I saw you maybe have something to add.
So something that I was just maybe pose to everyone as well is typically in terms of biosafety, we're governed by the institution biosafety committee, right? I thought that there might be a blind spot there where non-replicating system is not dangerous. Because in the biocontainment perspective, sure, the non-replication may not contaminate the ecosystem, but if they get into human, it can still be dangerous. Say a non-replicating, purely hypothetical, I have not seen the works that can do this. Say artificial cell or synthetic cell that are non-replicating, but get embedded into neurons. They are also non-replicating. Then that can be dangerous. But that, to me, is often the blind spot in the IBC, where they often think about replicating virus, replicating bacteria as dangerous, but not non-replicating artificial cell. If you say, I'm working on non-replicating artificial cell, then that kind of question may not appear in the questionnaire. So I don't know whether this is worthy of discussion, just a quick thought.
Michael, please. Yep.
I'll just pick that up quickly. I think that's a really important point, but it kind of changes to more of a chemical safety point, right? As in, these are mixtures of lots of chemicals, right? And so knowing what they are is very important, right? Knowing what they all do and how they all act is very important. And that's just a normal, I mean, for us, it's called a COSH assessment. I mean, probably something different in America, I don't know.
Juan, and then I'll go to Richard on our committee.
I'm picking up on something that John Glass mentioned earlier, which is making viruses. There is now a growing literature of groups which are trying to build, instead of using the type of systems we are building, they are using the same technology to produce viruses. And this is worrisome, of course. I think what you need to, because the number of chemicals that you use is very few. It's for our systems is 7 chemicals. That's all you need, and then you get all these complex behaviors. So what I am saying is that it is possibly what possibly needs to be done is to actually come up with some list of chemicals or of types of chemicals which could be used for this type of cells, and have them graded. And when you buy something from Sigma Aldrich, or you learn some synthesis, you need to register it somewhere. There has to be a registry somewhere.
I'm going to invite Richard Murray to ask a question of this panel.
Great. Thanks, Cameron. And thank you all for a great set of talks. So I guess I want to pick up on this point. Maybe I'll say it as a statement and then see if people push back on it. I think you've all said it in certain ways, right? There's certainly potential risks. I guess I want to argue and understand where this is missing, that the risk endpoints, human health, damage to the environment, et cetera, are the same, the thing that we're worried about where the harm might occur. And the pathways to getting there are also kind of understood, right? I mean, you can already produce something that is antibiotic resistant, for example, right? A gain of function or something like that. I mean, we already have, Michael mentioned it, Mike Booth mentioned it as, chemical safety protocols, right? You have to put a chemical out in the environment, right? We have processes around the world for doing that. Same thing with biosafety protocols, right? We have biosafety protocols, including with viruses and other things. So it seems to me that the existing mechanisms that we have for biosafety and chemical safety and the way that those get used in laboratories, but also in products, can cover synthetic cells. And so we don't need to do anything new with respect to synthetic cells. So I'm stating it that way as a kind of, blunt statement to sort of say, well, not really, think about, right? Here's something that we would need to do that's different.
Lynn, please.
I couldn't agree with you more, but re-echoing what John and others have said, these are even more wimpy than, the wimpiest ones we're growing in the lab, that they really are synthetic cells on life support. And so it almost puts them at an even lower BSL level. So until they're at the point that they can take care of themselves in a natural environment, I'm even less concerned about those.
I agree that the existing frameworks should be applied. The question, I think, comes in when you've changed an extant organism so much that within the risk framework, it no longer should be classified as that original organism, but should be something else. And I think, John, this was a situation that happened with your synthetic cell.
So our synthetic cell is technically a biosafety level 2 cell. But a number of countries have already said that it can be biosafety level 1 because of its inability to infect anything or to survive outside of its very complicated laboratory media. But to the thing Richard said, I will also add that the mirror life thing we've talked about is something completely different because it would evade all of our defense mechanisms short of fire and acid and such. And so that's sort of like beyond BSL-4, but anything else you make in terms of synthetic cells, I have no real concerns about any of it. Yes, you might be able to screw up and hurt yourself if you made something dangerous in the hood where you massively inoculated yourself with it. But for it to get out in the environment, I'm not worried.
Yeah, so let me ask on that one, John, on the mirror one specifically. So of course, bacterial overgrowth, infection, all of those things I think would be something we would protect against. But maybe you're right that we wouldn't have the right tools to evaluate that for mirror life somehow, right? Or just that, for instance, there's going to be something out there that'll kill this thing. And the question is, does the thing that killed the bug, is that worse or better to the human host or whatever it is? But here there's nothing, right? Is that, I'm trying to, that does seem to me to be kind of different, right? That maybe our existing frameworks wouldn't quite capture, because we didn't even know what to measure, right? Like, how would you measure the growth rates or toxicity or other things? Because the normal assays that we might use, for example, might use standard media, right? Put in rich media, it doesn't grow. Okay, well, it's not a problem, right? No, but that's not the point, right? The point is it could grow, right, in different media or on different things, right? Something like that. I don't know, I'm, asking the group, what do you think? There's a difference there that's like, yeah, we need to, again, from the point of view of our committee, think about adding on, right, some things that we look at as we look at the various things that might count as synthetic life, right? All the way from, non-replicating things that look more like chemistry, all the way up to things like mirror life.
So for instance, Rich, Steve Benner has long said that he thinks a better life would be made using 6 nucleotides, his system. And such a thing might be made, and in some ways it might be better, but I view it as, I don't worry about this because we have no idea how to make such a thing. And we're nowhere near it. Whereas mirror life is the exception, because we wouldn't, if someone started with a strain of E. coli that could grow on salts and glycerol, there would be and that ever got out of containment, there would likely be no way to stop it. No phage, nothing can eat it. And so it is so different that it almost needs a different set of concerns. Whereas ordinary life, the way we envision it, cells that are variations of cells we see, even if they do different chemistry, they're still going to be made of proteins and nucleic acids and membranes and things that are assailable by the stuff in our world. Ordinary synthetic cells, no problem.
I'd like to invite another member of our committee, David Gilliam, to ask a question of this panel.
I just want to first say to John that I appreciate the comment that I believe you said it was about 4 years ago that people were saying, you were talking about Mirror Life and the ability to do this. And it took several years for that to actually come about and say, oh, actually, there is a risk. And so I want to ask a question. This gets a little bit into the nitty gritty, but what regulatory guidance do you follow within your own space currently for your research on synthetic cells?
And I would just add on a little bit, since we do have Michael here, you know, we are interested in understanding some of the differences, maybe even between, say, UK, Europe, and the US. So I do want to also have that, you know, if we could have that as part of the questions as well.
So every research project that we consider at the JCVI, including the synthetic cell stuff, we have to take to an institutional biosafety committee, which has both our faculty and representatives from the community that will discuss this. And I assure you that our work to build our cells with synthetic genomes got a lot more discussion than anything else ever has. Before we even started the work, Craig Venter convened a panel of bioethics, legal, religious, safety people to discuss the risk of synthetic cells and whether such a thing should be built, or is it too much of a risk. And it was the decision of this committee that published its results in Science in December 1999 that synthetic cells pose no risk more than ordinary things as long as people are thoughtful about what they do.
Neha, please.
If I can chime in, we in our lab, we treat things at sort of a BSL-2 level, at a BSL-2 level. So, you know, what that means is you aren't dumping things down the sink. You aren't throwing things in the normal trash. Chemical waste, including vesicle waste, goes in chemical waste. And so I would just add that.
Maybe just as a kind of a follow-on question, you know, is as you're thinking about the, you know, the current development, but also even the future development of the types of synthetic cells or the chemistry involved in them that maybe are, I'll say, non-canonical, you know, what are some of those specific risks that you are starting to now evaluate that maybe are different than the current or existing frameworks? If that has started to come up as a conversation with your groups or even just the broader synthetic cell development community, I think we'd like to know, or we'd like to understand, you know, we understand that there are, you know, risk assessment frameworks in chemistry and other types of biologicals and biologics and living organisms, but, you know, what are some of these, you know, maybe it's a little bit speculative in some sense, but, you know, what is sort of also coming to mind? And then I'll also kind of, you know, even chime back to Elizabeth, the question about, you know, well, how are we going to, what do we need to do to be able to measure when we start working with different types of chemistries, or what kind of advancements should we really think about as we get into this newer stage of synthetic cell development?
As John said, I think that you need to know what you're working with and have a good assay for identifying release from your spaces. So it needs to be fit for purpose. In our laboratories, we're primarily BSL-1, BSL-2, and we're starting to think about how to monitor our spaces, especially as we think about using plasmids or using laboratory evolution that might result in samples that are maybe a bit more robust against naturally evolved life.
A decade ago, we had a project to look at the microbes in indoor and outdoor air. And the way we were collecting this, I collected the bacteria that we filtered out of the air into 1 molar sodium azide, thinking that this would stabilize DNA that we collected, but would kill anything that we put in it. And to my astonishment, when we sequenced the microbes that were in our 1 molar sodium azide, we found it was only one thing. And it was a Ralstonia species that had previously only been found in an Italian nuclear power plant coolant water. But the point is that unless it is extremely acidic or high temperature or basic or oxidative, you really, so organisms can be engineered that can live in almost anything. And it is critical that you really, as Elizabeth just mentioned, think about what you're building, because life has a way. And it is important to really think through what you're doing and consider what might happen, or do experiments to see, are your containment measures suitable?
Michael?
Yeah, I mean, I agree with this, with everything everyone said. I mean, answering a couple of questions all in one. I mean, I agree with Richard, right? And especially from the bottom up scale. I mean, for instance, for our lab, it's a chemical problem, right? We're not, and something John mentioned from a different perspective, right? We're not close to creating life from scratch, right? I'm not worried that one of my students will make a vesicle and it will be alive, right? Not even the slightest, right? So that's not a concern for me, right? We'll discuss other things in terms of chemicals and DNA sequences we might be expressing for other reasons, but I'm that way. I'm not worried for that reason.
Can I show a video?
Let's see if it will work. I'm going to look toward the NASM staff to see if that's possible.
Something that I couldn't show in the presentation. We are beginning to find out that if you start with the population of totally innocuous vesicles, and you feed the population with some, say, some metal that will be a catalyst for the photochemistry, we see that there is a tendency for some members of the homogeneous population to take up the catalyst, and then they become extremely efficient at eating or at self-reproducing. So this is a conversation which is happening, is taking place in our lab now. We call it the Darwin finches, whatever. These systems have the ability that whatever they find, they can actually use for their improvement, so to speak. So that combined with the chemical evolution, to me, poses problems. And I don't think that that this is only chemistry that we have to be worried about, or science of material science. I think we need to start thinking afresh in some areas about containment. I cannot offer any solution to anything, but what I can tell you now is that this is something we're discussing in the lab. Our Darwin finches, what the heck is going on? Why do they do this? What happens if they encounter the right food somewhere? What the heck? And they self-reproduce. It's not as if they are totally static. No, self-reproduce without any question. So I believe that we need to start thinking about something new.
I did get a note that Elizabeth will need to drop off, so I just wanted to, before she does that, I just wanted to thank her, and us and the committee are grateful. So we will just go ahead and let you. Thank you. I think there's well, okay, I'm actually going to go and pivot here to our chair, Peter Carr, for a question.
Oh, thank you. As we think about the mirror life issues, and I don't want to sort of overly focus on simply that because there's a broad spectrum of different kinds of synthetic cells we're talking about. But one question I did have is with the immunology argument with synthetic cells, because I remember research in the 90s on what were called retro-inverso peptides that were all D peptides, and part of the point of working on them was specifically to use them to stimulate immune responses. Now, I do remember that the responses to those peptides were frequently weaker than one would hope for, and that in particular, they didn't bind as well as hoped for to MHC2 complexes. But nevertheless, there were immune responses. So I think what I'm asking the question about, what I'm hearing people saying is that they're completely invisible. These mirror image peptides, proteins would be completely invisible to the immune system. I'm expressing skepticism that that's the case, but rather that they would be more weakly addressed by the immune system, if anyone would care to comment.
And I would just add, as I'm an RNA biologist by training, so we had Spiegelmers and all these other types of chemistries that show up in the RNA world. And I think it's this interesting question about how, well, one, it's like sort of do those concerns, show up, but also what should we be thinking about in the development if we start to leverage, these types of chemistries in synthetic cells, Juan, to your point, like kind of this different framework. So I'll pose that question to everybody here.
So as I guess the only member of our panel who was on the Mirror Life team, invisible is an exaggeration. It works well when we're talking to journalists and things. It is weaker, but I think it is important to remember here that as we know from the outcome of humans with weakened immune systems, is almost any significant defect in the immune system is a recipe for a life of woe, or more likely a really short life. And we know from experiments done with a variety of 200 amino acid proteins that proteases don't degrade them, and that we get almost no innate immune response. And without an effective innate immune response, you don't get much of an adaptive immune response. So what the immunologists that we have been working with tell us is that it is just something that we really shouldn't go there, although there could be real uses for these proteins if made abiologically as therapeutics because of this absence or near absence of an immune response that might make them more effective therapeutics.
Thank you for that information. That's helpful.
Even nucleic acids will generate a little bit of a toll-like receptor response.
Another question that it kind of goes actually to Neha's teaser at the end of your presentation, thinking through the combination of theory and understanding that's going to be an important part of synthetic cell development. And I'm going to ask a very big question, which is, well, there's still a lot of theory that we're trying to figure out just about cell biology and the origins of these types of cells. So maybe a question, and it's probably for everybody here as well, is like what are we missing still, right? We are in a sense starting to speculate about these synthetic cells, these replicating organisms, but what are we still missing? And maybe to even your point about the role of AI in this process, sort of what is this next step in the development of synthetic cell research that is on the horizon and or still lacking questions? This is for everybody, but I figured I would start with Neha because left us on a cliffhanger.
I can try to kick things off. I think what we need next are, like I mentioned, the design rules. And we need reproducibility and so that's going to require automation. It's going to and we will be leveraging AI to sort of figure out, okay, we mix these components together. What does that lead to? And can someone else do it? These are really important questions that we need to figure out. And then in terms of theory, what I mean is, you know, when we start to map components to function, we should try to, at the same time, be developing theories as to, are there general rules that are linking these two as we go? Otherwise, I foresee a future where people are just generating massive amounts of data without really taking the time to think about what it means.
Juan is in full support of that.
Complete and fully supporting that. The reason why I got into this type of problems is because somebody who in the audience, who people in the audience may recognize the name, and myself, we became very interested in understanding if we could apply physics to life. And we zoomed in. His name was Murray Gell-Mann, a very well-known physicist. So we started thinking, how do we apply physics to life? And then we found out that there is a ton of papers, a ton of models to discuss very specific problems, very specific behaviors in the cell or in ecosystems and so on, very specific, and they're tackled with physics. That's not a general theory. A general theory requires something like, for example, in Maxwell's equations, for Maxwell's equations and radio, you cannot be, Maxwell's equation provided the theory which encompassed everything about the electromagnetic, the unification of electricity and magnetism. We're missing that. We're missing such a theory. We're missing a theory that we can then fine-grain and go into the many problems that people can tackle. I think, and I will finish as quickly as I can. I think what is necessary is to actually start a new curriculum of biophysics. I think biophysics is Rashevsky raised to the 10th power. Rashevsky was somebody who did all kinds of problems, and it always was the diffusion equation or something like that, and then he solved all problems. It has to be, we need to have an impact on the curriculum of biology and to accommodate this type of thinking.
I'd like to go to another committee member, our chair, Felicia Wu.
First, thank you all for your terrific talks. Very, very thought-provoking. I'd like to build on the point that many of you have made, which is that in the absence of synthetic cells that are actually viable, what we're talking about really is kind of in the space of chemicals. Now, in chemical risk assessment and toxicology, what we typically say is that the dose makes the poison. And I am wondering if any of you can envision any scenarios in the future even with synthetic cells that are not viable, where they would build up to such an extent that we would start to have to worry about the doses that could cause harm to human health or to the environment. Would these applications be in agriculture, in medicine, in something else? And I'll give you an example. So there are some naturally occurring toxins like arsenic that are naturally in our Earth's crust, but we created PFAS. And now PFAS is everywhere. Can you see something like this happening with synthetic cells? Thank you.
I think Lynn has a starting answer for us. Reply.
Certainly, I'd like to get back to one of my philosophical points during my introductory remarks about life being an emergent property. And so I'm sorry, Felicia, it's sort of pushing back a little bit about the presumption of your question. And that is, if we really do make that leap from chemistry, from combinatorial chemistry to life, you will not be able to predict what you're going to find. So those interactions among the chemicals that were easily predictable pre-life, if life is an emergent property, you will not necessarily be able to predict them post when you've crossed that line. So I just wanted to make that point.
Michael.
Yeah, it's a great question. One of the important points to think about is most of the components, if not all of the components, unless you start thinking about some of the chemistry that we've been doing, are biological molecules. I mean, assuming you're not using mirror life things, right? So they are all, there are pathways in cells to remove and destroy and recycle every single component in a synthetic cell, right? If we're thinking about chemicals, right, there's situations of nanoparticles, for instance, that can be very stable and they can accumulate, right? So obviously there's if you start to use chemistry with them, you need to consider that, absolutely. But I think like the basis of them should be, and I mean, and this is partly what we should be designing them to be in the first place, is biodegradable.
Jimmy.
I still want to challenge the notion that non-replicating systems is not life and should simply be viewed as chemical systems. Because I think non-replicating system that can last as long as some of the cells in a human body should be regarded the same as those cells. But in the current, if we assess this artificial cell system or this complex chemical system as simply chemicals, then in terms of current regulation, we're not assessing them as like cells. And in human body, there are a lot of cells that are non-replicating, but they are alive. So I think that also occurs linked to one limitation in the current artificial cells is they usually don't last that long. But if theoretically, hypothetically, eventually, if we have artificial system that lasts long and interface with biological systems, whether in nature or in human body, then they should be assessed not as chemical system, but as life. Then that might.
I completely agree with you.
Yeah.
Thank you.
And then we are got a couple minutes left, so I'll go to David once again for one more question. Then John, I'll go to you. Sorry.
That's
David?
okay. Complete.
I just want to ask a question about, I mean, given the fact that currently we have a regime that is looking very closely at dual use research of concern and gain of function and like a high scrutiny on that and potentially canceling research, I'd like to ask you guys, how do you communicate what you're doing, the risk of it, like the science communication piece, so that people are comfortable with knowing what you're doing? How are you doing it at your institution? How are you doing it with the public and in your publications and so on and so forth? Because I do worry that people might interpret things like this as gain of function or something like that. So really, how are you communicating it, and are you concerned about any of that?
Well, in my case, I've found over the years talking to people that it depends a lot on the application. So I've never heard of anyone who's turned down insulin and angsted over whether it was transgenic or not. When I've been to the most anti-GMO groups in the Netherlands, when I talked about biomining and pulling metals out of spent electronics, they said, "Oh, if it's that, it's fine." So at least in my experience, it's very much application dependent. And if people really understand why it's good for them, or that how society can benefit, they say, "Oh, that's really cool, that's wonderful," and they don't think of it as gain of function.
We have 1 minute left, so I will make sure that if there's any last things to add. John, please.
So at a number of us on the call are members of the Build a Cell Consortium, this organization that has the goal of facilitating synthetic cell research. At the meeting at Stanford in September, there was discussion of coming together to try to propose to DARPA or someone to build a living synthetic cell. And the suggestion was, for instance, that its genome might not need to be very large to live in the lab. 20 years ago, George Church and Terry Forrester proposed a guideline for a synthetic cell. It was somewhere between 100 and 200 genes. And they said that this is what you would need for a cell. Now, in my minimal bacterial cell, we found that most of the genes encoded by that organism are present in everything, including 55 genes that are conserved in plants, animals, archaea, bacteria, and we have no idea what they do. And that really does say that we are a long way from being able to achieve some of the goals that we have been espousing in the synthetic biology community, the synthetic cell community, this idea of one day being able to build cells where we can interchange parts, where we can design these chimeric organisms to do remarkable things. We are still an unknown distance from really understanding how life works. The things that people are doing with non-living synthetic cells, they're certainly getting us closer. They are, but again, these are systems that in many cases people put together only with components that they know what they do. They're not living, and there could be remarkable tasks that are performed by some of these cells, genes of unknown function, or proteins of unknown function that are necessary for life. We've got a long way before we are really able to design cells that will solve a lot of human problems. But we should still do it.
Well, I am glad that you six, along with Dr. Elizabeth Strahalski and George Church, are working on these questions. I'd like to thank, on behalf of this National Academies Committee, thank you all for your time to speak with us. At the moment, we are finished with our first session of the afternoon. We will take a 15-minute break. We will regroup at 3:30 p.m. Eastern Time for the next session led by Dr. Emma Froh.
Thank you all very much.
Thank you.
Bye, folks.
Thank you.
Thank you all.
Thank you.
Good afternoon, everyone. My name is Emma Froh, and I'm an associate professor at Arizona State University and a member of the National Academy's Committee on Environmental, Biosafety, and Biosecurity Considerations for Synthetic Cell Research and Development. And I'll be moderating today's second session, which is titled State-of-the-Art Approaches to Biotechnology Risk Assessment and Mitigation. So thank you very much to the panelists who have volunteered to present today, and thank you also to those of you tuning in online to watch this session. So this afternoon session will focus on really trying to deepen our understanding of contemporary biotechnology risk assessment and mitigation strategies. We're going to examine state-of-the-art methodologies that can be applied or adapted to synthetic cells and other emerging biotechnologies to promote responsible innovation and enable proactive risk management. Special attention will be given to the biosafety, biosecurity, and environmental risk assessment dimensions of this work. Our discussions will encompass methodologies, regulatory perspectives, and practical tools for evaluating and managing risks across the research domain, as well as a range of biotechnology and potential synthetic cell applications like we were hearing about this morning. So for this session, we have a great lineup of 4 speakers. We have Rebecca Moritz from Colorado State University, Ben Trump from the University of Michigan, Richard Lenski from Michigan State University, and Greg Jaffe from Jaffe Policy Consulting. To make sure we have plenty of time for discussion, at the end of the talks, what we're going to do is save any questions for the panel discussion. Each speaker will have up to 7 minutes, and to ensure we have time for everyone, we will be keeping to this timing. So I'm going to keep the introductions very brief. If you would like more information, the speaker biographies for this session can be found on the event webpage. So with that, we will get started with our presentations. First off, we will start with Rebecca Moritz, who's the Biosafety Director at Colorado State University. So please, Rebecca, when you're ready, start us off.
Thank you. First of all, I would very much like to thank the National Academies as well as the committee for the opportunity to present today. So I am actually, I'm going to pivot. I was going to spend very little time on this slide, but the way the conversation has gone so far in this meeting, I really want to emphasize the importance of the timing of when the risk assessment occurs. So speaking from a biosafety officer's perspective, a biosafety professional, I could find out about any experiment anywhere on this timeline, when the experiment is being designed to when it is in front of the Institutional Biosafety Committee. And usually things will go much faster from experimental design to experiment initiation. The earlier in this timeline, someone like myself finds out about the research. And why is that? Because we can be part of that experimental design. That risk assessment is happening as the experiment is being designed, and we can actually help cut through the red tape and help that research start faster. So it goes through the Institutional Biosafety Committee in a faster manner. So I would actually start in reverse. I would ask the high-level questions before I got into kind of the nitty-gritty about the experiments themselves. So I would want to understand, what is the researcher's goal or purpose behind doing this work? What do they really want to achieve? And also, what is their hypothesis and what is their confidence in their hypothesis? Is this kind of a shot-in-the-dark experiment, or is this a set of experiments that there's a lot of data out there to really suggest that something is going to work? But then I'd also want to know, where is the funding coming from? And the reason I say that is because if it is government funded research, there might be certain regulatory requirements that are needed, as well as it most likely would have gone through heavy scrutiny on the ethics side during peer review. But if we're talking about private funding, slush funding, startup funding, that level of review about whether the experiment should or should not be done might have occurred or might not have occurred. So then going back to the experimental design, we went in great depth about this technology in the last session and what we are currently capable of. But I would really like to understand the components associated with the synthetic cell being made. I want to understand, what is the membrane? Is it a reassortant? Is it a chimera? Does it model everything that is already existing in nature? Or is it novel in some way, shape, or form? The same with the genome. What genes are there? What are the functions of those genes? And what do you anticipate they're going to do? Are there mutations? Are there chimeras? Are there reassortants? As well as the energy source for the cells. Is it going to be something novel? Is it something that already exists in nature? Is it going to biologically contain it to the lab in some way, shape, or form? And then probably one of the more obvious questions, can this construct replicate? Or is it replication incompetent? Obviously, that's going to dramatically affect the risk assessment. But then also start getting into the questions about, could this synthetic cell potentially outcompete wild type cells? Is there an evolutionary advantage? And that's something we might not have the answer to at the experimental design stage. But then also, what are the risks to humans, animals, plants, and the environment? Again, we may or may not know that answer. But then we move also into the procedural hazards. It's not just the components, but what are you actually going to be doing with the cells? What are the number of cells we're even talking about here? Are we talking about a small amount? Are we talking about a large amount? Also, where is the work being done? Is it going to be done in proximity to naturally occurring cells? Are they going to be sharing the same biosafety cabinet, the same incubator? Are the same people going to be doing the work? Do we have to, one, worry about contamination, but also potentially worry about horizontal gene transfers, right? But then also, again, this was talked about in the last session as well, is there some way to identify that synthetic cell versus a wild type cell? Is there some sort of watermarking, barcoding, or something else that will allow you to understand what the differences between a synthetic cell and a wild type cell are, and identify them? And then if those cells can replicate, are you going to allow them to evolve? How many passages are you going to have inside of the laboratory, or is it going to kind of be a one and done thing, right? Because obviously the cells are going to change over time. They're going to adapt. They're going to become more efficient. So from a biosafety perspective, taking all of that information together, what are our risks? What are we actually dealing with here? And there probably could be a lot of unknowns, but also there could be things that are not super risky. But oftentimes when there are a lot of unknowns, we will model things off of, what do we do now? Or we will also be more conservative in our responses. That's historically, if you have an unknown bacteria or an unknown virus, we oftentimes will use it at a higher biosafety level until we have a greater understanding what the risks are, and then we'll lower it to a lower biosafety level, for example. So from a biosafety perspective, really thinking about, what are the work practices that make sense for this work? What type of personal protective equipment are you going to need? So most likely your lab coats, safety glasses, and gloves. What type of facilities are we talking about? So most likely, at most, probably a biosafety level 2, unless we're talking about mirror life, and then that's a completely different story. Maybe even biosafety level 1, especially if it's replication incompetent and it's not going to be able to replicate, right? But then also thinking about, what are our administrative controls, our standard operating procedures, occupational health, because again, humans can move things around. While we might not get sick from something, they can stick to our clothes, our shoes. We don't always think of ourselves as the vectors of a disease that we can be. But then I also argue we got to think more about the biosecurity of the work, right? The material or the cells could be a target for theft. What if someone wanted to maliciously use this synthetic cell? What could the potential consequences be? Also thinking about the theft of the data and the intellectual property, as well as potential export control constraints that might determine who could actually be working on this project. And while historically work like this doesn't necessarily fall under the dual use potential, I think we definitely need to think about it. And the technologies, the things that we're coming up with, are they creating a roadmap for something else and for more powerful synthetic cells that could be misused? Now, taking all of that information together and really weighing the benefits versus the risks, should the research be done? It's usually not a question biosafety professionals necessarily get involved in, especially if research has federal funding and has gone through rigorous study section review. But especially if, like I talked about, where that funding, depends upon whether or not that ethical, discussion has been had. And, institutions really do need to decide whether or not they want to support that work being done, because they are going to have to invest in the infrastructure to be able to ensure the safety and security of the work. Thank you very much for your time, and I'm looking forward to the questions with this section.
Wonderful. Thank you so much, Rebecca, for starting us off. Next up, we have some remarks from Ben Trump, who's currently the president-elect of the Society for Risk Analysis, as well as an adjunct associate professor at the University of Michigan's School of Public Health. Ben, when you're ready, we'd love to hear from you.
And I think you're muted.
So I'm requesting control right now, and I don't see it. So I may just ask you to go next slide. I apologize. We'll go quickly through this. First, Emma, thank you very much for the introduction. So I'm Ben Trump. I am with the University of Michigan and with Tor Intelligence, and formerly from the U.S. Army Corps of Engineers. And to date, I'm going to be presenting some work. Click to start the remote control of the shared screen. I'm just going to keep going. There it is. Got it. Okay, got it. Apologies, gang. I thought I had this down. So over the last 15 years, um, uh, my colleagues at the U.S. Army Corps of Engineers, which I'm grateful for, Igor Linkov and Chris Cummings, my longtime collaborators, we've been trying to build risk assessment frameworks across a range of emerging technologies, from engineered nanomaterials to synthetic biology, and now increasingly synthetic cells. So this presentation builds from that experience. And I also want to focus on not just what we've done, but more on where the field is likely going, and especially where the systems we're evaluating are no longer stable, easily categorized, or even fully observable. So one of those core areas in biotechnology governance is something that we call the pacing problem. And it's the idea that innovation often moves faster than the frameworks that we use to evaluate and manage risk. This is not a temporary problem. It's structural. It happens with all technologies. By definition, there is lag time between when you're deploying a new technology, or at least conducting laboratory work, collecting data, and then developing standards for that. Gary Marchand has written about this for many, many years. But by definition, despite these problems, what we're seeing with biotechnology is that the gap between technology innovation and risk data collection, and especially the gap between risk data collection and risk regulation, is becoming wider, it's becoming faster, and it's becoming more consequential. So take synthetic cells. These constructs don't fall neatly into our traditional categories of living or non-living. Again, lots of discussion about this, but from a risk sense, that distinction is something that can drive us down one pathway or another for evaluation. Some may metabolize, they may sense their environment or perform complex tasks, but they don't replicate in the way that traditional GMOs do, or at least there are different concerns and considerations to be mindful of, hypothetically and potentially. Many are designed, or at least could be designed, using AI tools that rapidly explore genetic or biochemical design spaces far beyond human intuition. There's an awful lot that we can and cannot do using some of these different development platforms. And their applications are both vast and can often challenge or at least stretch the boundaries of the existing regulatory domain in the United States. So as a result, many of the questions that I've raised with it, throughout my work are very similar to those that Rebecca has equally raised. Is it containable? Is it reversible? Are there countermeasures? Do they exist and do they work? And most critically, can we quantify the risk in a meaningful way? So To answer these questions, we need more than just better data. We need better governance models, some that blend hard science with strategic foresight and adaptive governance. Some of the most effective tools that I've seen are what we call comparative governance models. These look not only at the organism or the product, but also how it moves through institutions, its function, how different stakeholders perceive and manage risk, what value does the product or the resulting potential product play in the marketplace, and how those perceptions affect deployment. So, and let me give you an example of some of the work that we've done to attempt to try and develop some numbers for this exercise. This is a fairly old example at this point. It's about 6-7 years old. So we had a team at USACE ERDIC in collaboration with individual stakeholders at FDA and EPA and USDA APHIS that worked on a virus, again, completely hypothetical tabletop exercise, but trying to develop a risk assessment for a virus-based biocontrol system for an invasive carp species. The questions we faced weren't about whether the virus works, but about how it might behave in a real world water system, with concerns and issues related to latency, to mutation, to interspecies actions, and horizontal gene transfer, among others. We had to shift from deterministic models where a single set of inputs leads to a predicted outcome to more probabilistic ones that could capture uncertainty and variability across multiple dimensions. I mean, to be honest, this whole exercise demonstrated how difficult some of this is and how much uncertainty that we have, but equally that many of our governance systems struggle to deal with that uncertainty range and to foster effective boundary conditions for it. And I'll skip ahead. So there's an additional layer that Rebecca also mentioned, which is how do we control information, or at least offer some protective guardrails or leashes related to the information that may go into some of these exercises. And it's becoming increasingly salient as we consider the use and convergence of machine learning and deep learning into this space. So risk isn't just about the biological product anymore, but equally about the process that creates it. That's a consideration that might be a unique emerging concern. Again, we're all aware generally of process versus product debate, but it might be a unique emerging concern in the United States because other jurisdictions already take a process-based approach to various extents. So some questions that we need to consider are what happens when synthetic cells are designed with LLMs or diffusion models? What about design platforms and when they're globally distributed and have open access concerns or demands? What about malicious or naive use that is limited not just to physical access, but when we have to deal with things like access to different GitHub repositories, various concerns related to that. So when we're thinking about distinctive integration, we have to ask the question, so where does this leave us? It means that we need to shift our mindset from viewing risk assessment as a one-time hurdle before deployment or something that at cost that must be borne by the system to treating it as an ongoing process of system management and product improvement over time. Approvals can no longer be static snapshots. Again, I know we look at pre-market and post-market issues, but trying to create some more process-oriented capabilities where we have improved iterative feedback, more adaptive processes that function at the interagency level is something that is critically important if we want to advance a series of products ranging from different biomanufacturing capabilities to considerations of environmental deployment. Just to wrap up, this requires some concrete steps. So one is incorporating some scenario-based testing, similar to what's been done in various other sectors to evaluate how synthetic cell systems respond to varying environmental, operational, or misuse conditions. Second is looking at considering open registries for synthetic cell designs and their functional components. Look at component-based risk assessment, enabling better traceability, monitoring, and shared oversight across our different agencies and at a bilateral and multilateral level. Third is developing oversight for artificial intelligence, deep learning, and machine learning, and other digital tools that generate or evaluate biological constructs. And last is investing in anticipatory governance capabilities across agencies so that regulation isn't just reactive, but proactively shapes development pathways. So last, we should also be considering how to better integrate risk and risk assessment early into the technology readiness level, or TRL, process. We've started to see some work related to this in the biosecurity field, but thinking at broader development, including safety, is if we're looking at early stage development, are there any questions or considerations or tests that could be done that give us, again, not a solution for risk, but at least early considerations that mirror the process of technological maturity as we proceed from TRL 1 to TRL 9 for government acquisitions. Again, if government is going to be a major funder of biotechnology research, we need to make biotechnology funding and development work in a TRL system that equally accounts for risk. It's not going to limit or hinder the process of development, but quite the opposite. Stands a good chance of easing the developmental pathway so that our regulators can better collect data early in the development process. We can overcome a risk-based catch-22, and hopefully foster a marketplace that's more constructive to a much broader diversity of product development. So with that, I'll close. Thank you very much for listening.
Thank you very much, Ben. There's a lot to think about in there. I'm sure there will be questions and discussion later. Third in our series of presentations this afternoon, we have Richard Lenski, who is the John A. Hanna Professor of Microbial Ecology at Michigan State University. Richard, it looks like your slides are up, so please take it away.
Great. So thanks for the introduction, and it's been a pleasure to hear all this incredibly interesting work. I'm very far afield in most respects from what's being discussed at this meeting. I'm an evolutionary biologist who works with microorganisms, but about 15 months or so ago, I became involved in these discussions over mirror bacteria. As by way of disclosures, I'm a co-author of the 2024 science paper and the associated technical report. I'm an unpaid member of an advisory committee for the Mirror Biology Dialogues Fund. And again, I would emphasize that I'm speaking as an individual, not as representing any particular group or institution. Let's see, where is the controls? All right, so I was also asked to comment on funding. So I have no funding that's related to this talk. It's strictly voluntary work. And by way of acknowledgements, there are about 30-some co-authors on that science report that are listed here. So it appeared in December of 2024, including one member of your committee, Kate Adamala, and two of the earlier speakers, John Glass and George Church. So mirror life, just to refresh, all organisms rely on molecules with chirality, that is a handedness that prevents strict superposition of one entity on another. That includes DNA, proteins, and more. Organisms use only one of the two possible configurations, but it would be in principle possible to produce mirror life that would have the opposite chirality. Synthetic biologists who are members of our committee suggested it might be created in the next 10 to 30 years. I believe George Church, in his talk earlier today, suggested that might happen even sooner. So why create mirror bacteria? There's the technical challenges. That's part of what drives science, is doing new things and the scientific curiosity about what might result. Also, mirror molecules are thought to have some potential therapeutic uses, in part because they resist degradation by the usual biological pathways. And one potential application then of mirror bacteria is that they could produce mirror molecules more efficiently than could be achieved by chemical synthesis. Let's see. Whoops. All right, let's see, I lost the slides. Can they be brought back up?
Just give me 1 second, Richard.
Sorry, clumsy with the mouse. Thank you. We can go to full screen. And a few slides forward. My standard control is not working here. If we could go a few slides forward, please. Doesn't seem to be working. Can we go a few slides forward, please, Layla? Thank you. I may have to just ask for advancements. Go forward, please. And one more. So importantly, our science article doesn't advocate banning the chemical synthesis of mirror molecules. What we instead proposed was precluding the creation of the bacteria. And this distinction, and it's been discussed in many of the previous speakers, rests on the fact that organisms, unlike chemical molecules, are potentially or actually self-replicating. And synthetic organisms with extreme and plausible risks therefore pose a special danger. Next slide, please. So there are two reasons, I think two predominant reasons of why not create mirror bacteria. First of all, as John Glass discussed, particularly in response to a question, mirror bacteria would likely evade many immune responses, and thus they could become opportunistic pathogens of humans and other multicellular organisms. Second, they would likely avoid most predators, and thus they could spread through the environment and disrupt both natural and managed, for example, agricultural ecosystems. Next slide, please. So the reason they would evade immune responses is because immunity depends on chiral interactions. And as again, John Glass alluded to, infections of mirror bacteria could prove devastating, even lethal. And the best information from the immunologists on our committee is that these effects would be similar to inherited immune deficiencies. And I think it's important because I sometimes get asked this question. It's not as though it rests on specialized virulence factors being engineered into the mirror bacteria. To initiate the infections would simply require that the organism enter a mucosal surface or potentially an abraded skin surface. And we know less about invertebrates and plants in terms of their immune systems, but what we do suggests they would also be likely vulnerable to mirror bacteria. Next slide, please. The other problem is, again, that they could spread through and disrupt ecosystems. Many bacterial populations in nature are kept in check by viruses and various predators like protists, other bacteria that are predators, nematodes, and so on. But these predator-prey interactions also rely on chirality of the predator to recognize and infect or consume the bacteria. Next slide, please. So it's almost certain that, for example, a phage, a virus that infects bacteria, would be totally unable to enter and replicate within the reversed machinery of a mirror cell. There might be some predators that would kill mirror bacteria, but importantly, the role of predators in controlling prey populations is not that they kill things per se, but that they gain nutrients and thereby replicate and increase their numbers in response to an increase in the number of prey. And so even predators that killed mirror bacteria would likely obtain little nutritional benefit, which would limit their numerical response and their capacity to exert effective control. So without these top-down controls, mirror bacteria could expand, their populations could expand, they could spread. Once their populations get large, they would certainly evolve, and potentially they would diversify into another tree of life, into diverse environments, performing new and different roles in those environments. So the conclusion of our committee was that these could potentially cause massive environmental harms through extinctions, degradations of habitats, and effects on nutrient cycling on top of the health risk to humans. Next slide, please. So it came up, a very nice discussion by several people earlier. Wouldn't these as synthetic organisms be poor competitors? Or aren't synthetic cells in general poor competitors? And I think that is generally true, that organisms that have been domesticated, that have been deliberately disabled, will generally be poor competitors. And in case of mirror bacteria, they couldn't consume many of the normal nutrients that their natural counterparts could use. However, there are achiral nutrients that mirror bacteria could consume, and bacteria can use or evolve to use some nutrients with opposite chirality from what is considered the standard. Next slide, please. And so what's important here is the balance between birth and death. The growth rate of a population doesn't depend on whether the organism is a good competitor, but whether its growth rate or birth rate exceeds its death rate. So imagine the little bar chart on the left, a stable population, some bacterial population in nature, maybe it cycles seasonally or whatever, but over years and decades and centuries, birth rates and death rates tend to be well matched. Otherwise, you have an invasive species or one going extinct if they're out of match perpetually. Now imagine a poor competing mirror bacterium that escapes into the wild. It may have a much lower growth rate than many corresponding bacteria would have, but it would also almost certainly have very little death, at least due to biotic causes. And so as long as the growth rate is greater than the death rate, the population would expand, potentially reaching vast numbers, even if they reproduce more slowly than their natural counterparts. Next slide. So what about containment and control measures? So the problem, and again, this was discussed actually in a couple of the preceding talks by Ben and Rebecca. So biocontainment could be circumvented. It could be circumvented by malicious actors, but also I would worry about ill-informed commercial actors who might want a more robust form of something to synthesize these mirror molecules. So potentially it would be fairly easy to engineer a mirror bacterium that could actually consume the natural and abundant form of glucose in nature. Physical containment risks human error and could also be evaded. There are some antibiotics that would likely be effective against mirror bacteria, not most, but other new antibiotics and countermeasures could be developed. Next slide, please. However, there are two problems with that. First of all, if we're talking about the context of human health, providing treatments globally and equitably during a pandemic would be extremely costly and difficult, if not impossible. And once something has escaped into the environment, if it's causing harm, whether to humans, other animals, plants, or whatever, once it's established, it would expand, spread, evolve, and diversify over time. And I know, again, in the previous talk by Ben Trump, he mentioned something that got me thinking about time lags and this emphasis on screening for escape. Once an organism that can establish in the wild has escaped, I think it becomes very difficult. There's a time lag between its escape, its detection, and employing countermeasures. And I think it would be game over in terms of bringing mirror bacteria, causing them to go extinct if they were to establish in nature. So I believe that's my last slide. Thank you very much again to the organizers and to all the other speakers.
Wonderful. Thank you so much, Richard. And fourth in our session today, last but not least, we have Greg Jaffe, who is the president of Jaffe Policy Consulting.
Thank you, Emma. And I thank the committee for inviting me here today to give my sort of thoughts on this topic. I do feel a little out of place here. I'm not a PhD scientist. I'm a lawyer, a policymaker, and a regulator. And so I'm really going to come at this topic from that perspective. I've spent the last couple of decades of my career working in the genetically modified organism area, genetically modified plants, animals, microorganisms, and so forth. And so that's sort of where I come to the topics that the points that I'm going to make today sort of come from. And I know that's not exactly what synthetic cells are and where they might be going, but I think it gives us some sort of guardrails for thinking about risk assessment and how that relates to policy and regulations. So I took control, but then, oh. So I think, some of the other speakers have talked a little bit about this, but I think when I think about risk assessment for a different product or a synthetic cell or a genetically modified organism, one needs to think about what is the purpose goal of that risk assessment. And risk assessments can have different scopes, and that's sort of what I want to talk about in my few minutes here is how do we define what that scope is? I mean, you could have, I think as Rebecca was talking, you could be talking about just conducting research. And so what is the risk assessment around that? And a lot of that talks about discussions that have been had earlier about risk mitigation. If you're in a a highly contained environment, a P2, P3, P4 lab, well, you may not have much of a risk assessment associated with that. You also might be doing a risk assessment to, talk about the whole process of developing an organism and then releasing it and a product far down the line and designing your mitigation management strategies for that. But you also may have a risk assessment purely to comply with regulatory requirements. And this is, I think, a different animal, because when a regulator is looking at a product, they're looking at it through a lens of what is their legal authority, what is their statutory authority, what is required of their regulations. And so that is usually much more narrowly focused on what there is. And so that risk assessment might be particularly narrow. So for example, USDA regulates genetically engineered plants, but they do it under the Plant Protection Act. And so they're really only looking at plant pests and noxious weeds risk issues. They are not looking at other things. They are not looking at worker safety, for example, or worker health. They are not looking at environmental releases into water bodies or something like that. They're looking at what is their legal authority and what is the decision they're going to need to make. And I think that's a very different kind of thing. And so when you're thinking about your risk assessment, that's what you may be thinking about in those instances, is how am I complying with the regulatory requirements? And so the second issue is, what issues are to be decided in that risk assessment? And again, I think part of this depends on what is the stage of development. Are we talking about just containment in a laboratory, production in an enclosed facility, release in environment? Those are going to have very different scopes for the risk assessment depending on what is going to be the likely use of the product or the cell or whatever you're developing is. And then you have to think about the issues. Is the risk assessment, as I said, limited to environmental issues? Is it going to look at food safety issues? If it's a food that's going to be eaten or a feed for an animal, is it going to look at worker and human health concerns? Again, a broad risk assessment about whether one wants to produce something or move down that line might look at all of these things. But if you're looking in the regulatory context, those things may be very limited. You may only be looking at one or two of those issues, not necessarily all of those issues. So another issue to think about, and there's been a little bit of discussion about this, although maybe so far in this webinar it's not been explicitly talked about, is do you only look at the risks or do you also look at the benefits? Many products may have risks, but they also have benefits. And so in some instances, we may look and say, do the risks outweigh the benefits or do the benefits outweigh the risks? So some people think of risk assessments only looking at what those risks are, but I think there's been discussion here today about not just the risks, but also those benefits. And the example I give in the policy or regulatory space is environmental versus food safety. Most of our environmental laws and most of our environmental regulations really do envision the risk assessment or the assessment they're going to do to approve a product is really a balancing of whether those risks outweigh the benefits. You might think about producing a new pesticide and that's going to get approved. Well, they may look and say, well, what is the pesticide that it's going to be replacing? Is it going to replace a more dangerous pesticide? Because any pesticide has some risks associated with it. It's intended to kill a pest. It will necessarily potentially harm other organisms out there in the environment. So the question isn't whether there's risks there, but are those risks outweigh that benefits or not? On the other hand, in the food safety context, it's pretty hard to find situations where we do a risk-benefit calculation. If a product is a potential allergen, the fact that if we all ate it, it might reduce the amount of heart disease in the country, or add the quality of life or extend life expectancy by a year. We don't tend to balance those. We don't tend to say, well, yes, it might result in a severe allergic reaction for 5,000 people, and some of those might end up in the hospital or dead, but we're going to increase the life expectancy of everybody who eats this food. We tend to say if there's a potential allergenic risk, we don't go any further. We decide there are other foods, other substitutes that we have in the marketplace and not to do it. So, and then one of the other things is, what is the cost of not implementing the technology, keeping the status quo, which may, as already has risks associated with it, because everything we do in life has risks associated with it. Nothing is sort of risk-free or completely safe. So that gets to the question of, is the product safe? And there's been some discussion of the word safety, maybe not as much in this webinar so far, but I wanted to make the point that, safety is really a relative term. I think there was some discussion, but some of my previous speakers on this particular panel, of uh, how risky do you want to be, or what's the level of risk? And so I think of safety as one, you have to have it as, again, every activity we do has some level of risk associated with it. Everybody ate lunch today, and eating lunch, you took a risk. That food could have been contaminated. You could have had a problem. If you go in your car, every time you go in a car, there's a risk associated with that. So we have to think about what is our comparator here, and what is that the level of safety that we're comfortable with. For genetically engineered organisms, they've come up with this compared to the safety of conventional counterparts that already have a safe history of use. But that doesn't mean that those current products have no risks or no impacts, because they do. A biopesticide, currently still has risks associated with, still has impacts associated with it. But we have either chosen to allow that to happen. We've chosen to take that level of risk in doing that. So I wanted to say that you have to think about what's your comparator here, what's your level of risk, and I've also thought about this in the genetically engineered plant or animal process, in the sense that everybody's made big distinctions between the way the US and the EU have treated these products. But if you look at the risk assessments, they've really come to the same conclusions. It hasn't been that their conclusions have been different, but those countries and their policies impose a different level of safety upon those. The US might say, well, there's a chance of something happening 1 in a million. We're willing to, 1 in 10,000, we're willing to take that risk. The Europeans might say, oh, we are not willing to take a risk anything greater than 1 in a million. And so it's not the issue of the risk assessment, it's the issue of the safety that people are thinking about. And so that gets to another point that I wanted to make, about when we're thinking about risk assessments and what you're doing. It's sort of the nice to know to need to know kind of thing. I think scientists and some people think that you have to answer every single question or have an answer for every single pathway and so forth. And that may be true if you're doing a broad societal risk assessment. But when you're talking in the regulatory context, it's really, what do we need to know to answer the particular questions that the regulator needs to do based on the law that they're imposing. It's not necessarily the need to go to every issue that might come up, but just the issues that are relevant to them. And I also wanted to mention, therefore, the product versus process distinction that I think Ben alluded to. I'm not sure I completely agree with you, Ben. I would argue that the US system claims to be a product-based system, but actually much of the risk assessment and much of what's done really takes the process into consideration. As an example, if you've genetically engineered a plant and you've added a new gene, we look at that and we say, does that, that new gene that produces a protein, is it an allergen or something? We also look and see, is that gene stably introduced and things like that. Many of the questions we ask are really based on the process, not the final trait or the final product that we've identified. And so I think that distinction, my view of this is that, you can't do any kind of risk assessment without looking at both the process of how you got there, because that informs the risks that might exist in the final product that one's looking at. And then there are some other considerations I just wanted to mention. I think that, one way forward is to think about harmonizing these risk assessment procedures and standards. Consensus can be very helpful, whether that's on the data that's needed for the risk assessment, whether it's the issues that need to be addressed, what are the risk pathways that we all agree are the ones that need to have answers to that risk assessment process. We've seen historically, organizations, international organizations like Codex Alimentarius has a number of documents on how you do the assessment of genetically engineered plants, animals, and microorganisms. OECD has done things. I think those are always helpful for cost, for timeliness, for a host of number of reasons. They do take a long time to do, so they're not there are problems associated with that also. I think transparency is critical in these things. I think, a good risk assessment will tell you what do you know? What are the facts that are part of that risk assessment? What are you assuming in that risk assessment? Because all risk assessments have assumptions associated with them. And then what are the basis for those assumptions, and how might the answer be different based on how those assumptions are thought about differently? Um, in the GE organism things, I think we've had a lot of success with public sector risk assessment research. There's been funding for decades at USDA for risk assessment research, where they funded, academics to go out there and think about the risk assessment pathways and exposures, and what data would be needed, how you would assess those. I think that is very good for one, the regulators helping get the questions they need answered, but two, for public acceptance, for giving public confidence here that you have that. And then finally, public engagement. I do think that, when thinking about these things, you have to bring in broader than just the scientific community, than just the peers, because I think that in the end, these are products that will affect a broader segment of the population. And so you need to bring those people along and get their thoughts, even if they're not have the same scientific knowledge about the particular things. So I'll end there. And I have no funders or sponsors for the work that was discussed today. It's really based on my many years of experience working with USDA, EPA, FDA, and regulators all around the world, and the thoughtful interactions that have influenced these observations that I've given today. Thank you.
Thank you very much, Greg, and thank you very much to all four of our speakers. And in particular, you've all done a fantastic job of keeping to time, which means that we can really have a full discussion with our panel. I think we have until we have 50 minutes, so until about 5:10 Eastern time for our panel discussion. And for this discussion, we'll have, I think, all four speakers joining us to take questions from the committee and from the audience. So I would encourage our committee members to put their hands up if they have questions, and also for those of you participating in the webinar to put your questions in Slido. But as we sort of get started, maybe I'll just sort of start with an observation and a kind of a question to all of our panelists. So I really appreciated that you're all coming from different kind of institutional positions, and really gave us both some granular and some really high level perspectives on thinking through sort of risk assessment and governance. Our particular committee is charged with thinking specifically about synthetic cells. And so what I'd really love to have maybe from each of you, from your vantage point, is some pointers, some guidance from you based on your positionality about what you would really encourage us to pay attention to when it comes to synthetic cells. So in terms of perhaps any characteristics of synthetic cells that you would really encourage us to focus on, any particular governance gaps that you see from your positions. We've heard several things have been raised about coordination across different agencies, about the living, non-living distinctions, about comparator organisms, all sorts of things. I'd really just sort of love to hear from you about, when we're thinking about effective risk assessment, and from your vantage points about what works and doesn't work well in your current positions, what would you really encourage us to attend to?
I'll go first. I'll bite it off first for everybody.
Thanks, Rebecca.
I think one of the hardest things is regulation is never going to keep up. The technology is moving so incredibly fast. But that also means the practitioner, like me, it's incredibly difficult to keep up with that technology. So what training and education is available for that biosafety professional in the field? But then also on the flip side, the education that we provide to researchers. We teach them how to become scientists. We don't necessarily teach them, in those fundamental classes, how to do biosafety and biosecurity and how to incorporate it into their science, right? We had some really great conversation earlier today with John Glass is a great example of the things that they've done at JVI, where they have incorporated that into the conversation. But as this technology gets more commonplace and there's a lot more people working on it, we're not going to have that same level of attention as to the safety and security, as well as the just because we can, should we conversation.
I'm happy to go second. So I think it echoes something Rebecca said and that all the other afternoon panelists have said and that John emphasized early on is to have very broad discussions. I think that's incredibly important because the goal, at least for me, should not be about dotting I's and crossing T's. It should be thinking importantly about what serious risks are. Stepping way back in time, I was involved in some of the early discussions for an Ecological Society of America and then for a National Academy panel on genetically engineered organisms in the environment. And I think sort of a consensus that most of us reached on that, and there's a report, I can't remember all the details, was that it's a combination of the risks of an organism establishing and what adverse effects it might have. And we tended to have the committee for the National Academy, at least, kind of split into two subcommittees because the plant biologists and the microbiologists had rather different perspectives on this. I think when a plant escapes or some of its pollen gets to another field, it's thought to be something that can be addressed fairly straightforwardly. But if there's severe risks with a microbe because it's a pathogen or because it's a novel form of life like mirror bacteria, and this comes to what Ben was talking about, there's a lag in all these processes, and there's certainly a lag between escape and detection, and between detection and harm, and between harm and mitigation. So I think the you need to think both about the establishment question and why you're worried about that establishment. And it's the combination of a potential establishment with potential severe risk that I think is where special attention needs to be. That's not to say that other issues of human health and so on aren't important, but I think as an environmental biologist, an ecologist by training, evolutionary biologist by practice, that's the most worrisome scenarios.
To try to answer your question, I would agree with Rebecca that regulation and policy is always going to play catch up. And I think that was Ben's slide, and I think that's very accurate. So one comment I would just make is that it's very important to keep policymakers and others in the conversation and really bring them along as the technology or as the processes are being developed and moving forward, to try to lessen that gap. It's going to be difficult to do, but I think you have to do that. The second thing, I guess, in thinking and hearing, I've been listening all afternoon, is a lot of our laws and our regulations are very siloed. They really deal with, a specific topic, food safety or worker safety or plant safety or other things. And some of the risks that Richard was talking about or others about, releasing an organism that could, that would, that humans wouldn't have resistance to or something like that. I'm not sure where that comes into play. I mean, NIH regulates the research, but if we've passed the research stage, I don't know where that goes. And so I think one needs to think about whether there is new policies or new things. And then you get into some of these questions which are more ethical. Should we do it or not do it? The government has not generally played in those activities. Those have been societal in different ways. And some people, I think in the biotech genetic engineered area, tried to bring those in to the regulatory system, and it doesn't fit very well. They tried to mask safety concerns, mask, ethical concerns as a safety concern, but that didn't work. And there were societal concerns. Should we, engineer this or do that kind of thing? So I think we have to have other ways to engage on those issues.
So just something to add quickly, and just two quick points. A idea that Greg brought up is, again, what is nice to know versus what do we need to know. To tie this back into the governance gap debate, it's when you have that lag time, you need to make a judgment fairly quickly about when you see something coming up, what are the list of parameters, like hazard, what are the hazards that we need to characterize? What are the exposure pathways that we need to be worried about? What range of consequences do we need to be very precise in our assessment or relative? And what level of outcome is acceptable or unacceptable from a risk tolerance and risk perception issue? The challenge that we're facing is when we're dealing with these gaps, we have a gap both in how information is being received, and both how it's received, who is dealing with it, how it's coded, etc. But equally, there is an increasing delay, and this is where, from a regulatory policy perspective, why things get really bogged down, and I think is probably particularly problematic in this case, is we don't know what counts, or at least what we should be prioritizing in terms of where we should be investing more money and more attention into developing accurate safety assessments. So another component I wanted to bring up quite very briefly is, again, going back to process and product, because I think it's at the heart of this issue. Greg, I absolutely agree with you. Like, of course we cover process in the United States, but to your point, it's process gets captured in a lot of these institutional silos in product-based assessment. So it's while it's there, the fact that these silos exist often slows down the collaboration across multiple agencies, that where we could evaluate process at the margins or dealing with some emergent behavior and emergent conditions of new science, new and emerging science like synthetic cell. For me, it's not that that's an insurmountable barrier, but it's certainly adding to the lag on the regulatory policy side, that's limiting both our ability to calculate and benchmark risk, again, hazard exposure effect, as well as to identify, again, what does matter and what do we need to know right now in order to figure out if something is acceptable to go to the marketplace, or at least is acceptable for release scenarios, and what is unacceptable, and what are the conditions that might make it acceptable. I'll stop there.
Thank you. It looks like we have some questions lining up from our committee members. So Felicia, your hand is up first on my screen. Would you like to go ahead with your question?
Yes, thank you, Emma. Thank you to all of you for terrific speakers. It sounds as though just within the last few minutes, you all seem to agree on the gaps from a regulatory standpoint, and that risk assessment should not just be left up to regulatory scientists. And you know, I myself had worked at EPA 24 years ago when we did the risk assessment for BT crops, and the only reason we could look at those crops is because they incorporated protectants. EPA couldn't evaluate glyphosate tolerant crops because there was no plant incorporated protectant. And so those environmental issues did not really fit in anywhere, I think, to one of Greg's points earlier. I would love to hear your thoughts about whether there ought to be some sort of independent risk assessment body from a government, you know, as a governance structure that ought to inform the different regulatory bodies, whether they be EPA, FDA, or USDA, in terms of, as Rich was saying, focusing on the real risks rather than focusing narrowly within FIFRA or TSCA or PPA. Thank you.
I guess, Felicia, I'll respond by asking you. So let's assume we had some independent body that did a full risk assessment, as you say, addressing all the potential risks and things like that. The problem would be is what happens if they come up with risks that we don't have any laws to address those risks? Yes. In other words, you could go through all of that, but again, if there was a risk that was associated and there wasn't a proper legal authority to deal with that, I don't know what we would do in those instances. So people might argue that you'd be looking at that would be a waste of resources, because you'd be looking at things that if the purpose of this is for the government, then they'd be looking at things that the government has left to the market or to some other situation. It wouldn't be a good use of the resources to do that.
I understand your point, Greg. I think that there are some stopgap measures. Aside from creating new acts, there are possible mandates that could be created in the space that, in the event that such risks were found. But I do take your point. Thanks. It's not easy to create a new FIFRA, for example. I think Rich and others had a point too. So, yeah.
Yeah, I was just going to say, I think, again, the example of John Glass going to a relatively narrowly focused group and having tremendous enthusiasm for mirror bacteria, and then when it became more broadly discussed, reminds us of the importance of the individuals conducting that research and the individual communities of which they are a part. And I thought in Rebecca's talk, she emphasized something that really resonated with me, that the biosafety officers also should sort of look holistically as well as at the details. So I think she suggested sort of starting, what's the question? And I think that's really important. And maybe, even just sort of instilling the value that the biosafety officers also, and everybody who's exposed to the discussion of a project, sort of say, well, is there something really weird that we have overlooked? That's not a law, that's more of an individual kind of responsibility, but I think it's an incredibly important part of avoiding hazard.
We also, well, we did. I'm not sure if we still do. We do have the Next Track Committee that is an advisory to NIH's Office of Science Policy. And they're supposed to be looking at emerging technologies and providing recommendations back to NIH about a lot of what we've talked about today, environmental spread, various risks, although I'm not 100% sure if that group is still a group.
I mean, just a quick add-on is a key challenge that we see is that coordination, especially when you're dealing with some of the more emerging components of synthetic biology and emerging biotechnology, is that the coordination, again, while there are individual agencies and institutions have decades of experience and very well qualified people working in and running them. Again, I've worked with many of them, much respect. Coordination across those agencies, it's not that it's weak, it's that it's not necessarily as incentivized as it might be, right? So again, we talked about EPA, we talked about FDA, we talked about USDA, APHIS, and there are others, including emerging players for dual use concerns. But as an example, like let's take a look at synthetic cells, right? Synthetic cells don't fit neatly into one agency's jurisdiction, right? A construct designed with an AI tool in Europe that is built with a cell-free kit in the US and deployed in US agriculture might touch 3 or 4 or more different regulators, but none of whom have complete visibility or shared data, if such data even exists, to touch at all elements of that problem. The US has faced a similar issue before, completely unrelated to emerging technology, but with the fragmentation of security intelligence in the aftermath of September 11th. Right. So again, among many, there are many reasons why the Department of Homeland Security was created, but it did serve as a useful vehicle to share information and help drive coordination in a very critical area of American public policy. So do we need a cabinet level representative to engage Felicia's thought experiment? I would argue probably not. But to Greg's point, you certainly need one that's grounded in hard law, because right now, if it's not mandated in hard law, again, executive order or a legislative act of Congress with specific responsibilities and even statutory authority, it's going to not necessarily have the teeth or drive the incentives to get all of your different agency players bring their expertise into a common toolkit and drive strategy going forward.
Great, thank you all.
David, would you like to go ahead and ask your question?
Sure. Thanks, Emma. Thanks, everybody, for being here today. Earlier today, we heard a lot of the scientists who are doing this research talk about things like containment and barcodes and the possibility of, keeping it in the lab, essentially. So I'm going to pose to you, since I think you're all risk assessment people, essentially, how would you do a risk assessment for an experiment that one of them talked about this morning, such as potentially like a biosensor agent or organism? I'll stop there. I'll just keep it like that and think about how would you do the risk assessment? What would you think about? And keep in mind, maybe in this example, it has an ecological risk. How would you do the risk assessment for that? If anybody would like to try to take that on.
Okay, nobody wants to go, I'll go. So if we're talking about ecological risk, now, David, in your scenario, is there a potential for human or animal risk? Or are we only worried about ecological?
Sure.
Sure. Okay. So thinking about it, a lot of the time researchers will be like, okay, this doesn't make me sick, this doesn't make animals sick, but they often forget that they are a vector of disease. So the closest analogy would probably be a plant pathogen or a plant pest. So you would have to think about how your work practices are going to spread it, how the individual doing the work could spread it. So you might have to do change of clothing, with stepping in foot baths, using separate boots, deconning all of your equipment coming out of that facility, thinking more along about it, those lines, right? So, or even like African swine fever would be a great example too, right? Something that would be decimating to the swine economy in the United States if we had one case. So even though it's a cell or a different organism, thinking about it along those lines.
Anyone else want to try to add on to that? I'm just thinking because, we talked about, I was thinking about the barcodes and I'm not sure how that would help except tell you it's already escaped containment. So I'm just trying to think through, if you're a person offering them advice on what to do, how do you tell them, before that you need to have the barcode to tell? You know what I mean? I mean, how do you do that risk assessment? How do you know?
It comes to what we had talked about earlier. When in that timeline are you getting notified, right? Is there actually that collaborative relationship between a researcher and the risk professional that is doing that analysis? And that is very much informed by their experiences with their institutional biosafety committee, their biosafety professionals, their risk analysis people, right? And also, no two IBCs are the same, right? So you would need to have somebody in that mix that's doing the risk assessment to say, hey, why don't you code this in some way so you can identify it later on, right? I mean, it's very much people dependent and expertise dependent.
So I'm
David,
not a risk assessment person at all. I'm a huge outlier at this meeting, but I would say nowadays, I mean, the whole genome is essentially a barcode of anything that's created. It's amazing what you can do by digging through lab notebooks and sequencing genomes and figuring out who gave what to whom. So in some sense, I think the focus on trying to track it and is it leaking, I'm much more concerned about the downstream consequences. So if you're talking about some sort of biosensor, where can it go wrong? And I mean, unfortunately, that is a really hard question. It's a harder question. I think one reason maybe people want barcodes is it makes it sound easy. Oh, we'll put a barcode in it and we'll swab. And that works great for radioactive spills where you kind of know the concentration and so on. But once an organism escapes that's self-replicating, you've already lost the battle if it has a serious consequence. So I would not put too many marbles in the barcoding and detection of leaks basket. I would focus much more on consequences, as difficult as it is, as many speakers have mentioned, to even anticipate those consequences. But that's where the harm comes in.
Real quick, David, so to your question, so our group, again, completely hypothetical, but about 7 years ago did try and build a risk assessment process for a virus-based biocontrol system for an invasive carp. Again, case study being we have Asian carp that are an invasive species that are going through the Mississippi River and potentially reaching the Great Lakes. And the question, again, tabletop exercise, that we were trying to ask was, again, how would you develop an environmental risk assessment for a case like this and the different pathways that you could execute invasive species control? And at least for this one particular case that we published, I mean, there is an easy answer and then there's the impossible answer, right? So the easy answer, well, not impossible, but very, very difficult. The easy answer is, again, you do your traditional risk assessment and then have a unique compartment in your risk assessment that looks at synthetic biology or genetic engineering's modifying factors, right? So what are the unique elements that you would not traditionally ask and an ecological or microbial or chemical risk assessment that relate to the modification that you're trying to take. Again, one of the things we were trying to assess is horizontal gene transfer. Like how easy or difficult is it to collect when you're considering a certain media, right? So we're looking at carp transfer via water, and then that carp would then be potentially taken and consumed by a person, right? So like what level to the umpteenth millionth degree are you looking at there to try and find a number there? So that's the relatively easy answer. The hard answer is in trying to create a hazard and exposure assessment capable to do this, right? You're looking at a degree of ranges, but that involves constructing a dose response curve essentially with very little information, and that's where you get hurt quite a bit. Because the kurtosis of your study ends up being quite broad, right? So your error bars and your standard deviation is going all over the place. So we tried to follow the path down that line, and I'm happy to send you that paper a bit later, where we were looking at the concentrated viral load and relative uptake and interaction effects, and expectations of efficacy of the modification, and its relative environmental persistence, and then taking that into again whatever object at risk was consuming that carp. So we were able to derive a formula to calculate that, but that formula implies a series of uncertainties and ranges that the risk system, for at least for environmental release scenarios, isn't in the United States isn't quite comfortable yet. So in terms of trying to figure out what do we need to know and what is nice to know, it's hard for me to make a distinction yet between the two of those.
Richard, the only thing I was going to add was, it seems to me the barcode only answers one question, which is where is the organism? And it's only helpful if then knowing where the organism is, one, you either have a method to contain it or kill it or whatever, or knowing that the organism has escaped, you have an anecdote, something that would then protect people or whoever you think the new risk is from, whether that's, give them a shot or, you could envision right now we work with, organisms that aren't engineered in P4 labs. And if it gets out, people, you're going to take your workers and treat them a certain way so they don't get sick or something like that. So the barcode is helpful to answer the question of where it is, but it doesn't answer the more important question is what's the risk mitigation that, and if there's no risk mitigation, if all the barcode does is tell you where it is and there's no second part, which is I think gets to Richard's point, then it's not particularly helpful at all.
Agreed.
May I ask my next question? Okay, so earlier, Richard on the committee mentioned or made a statement, and I think it was to get a provocative response or a response, is that, won't the existing biosafety or chemical safety systems be sufficient to capture this? How would you answer that?
I think it's questionable whether they would be. I think part of it is we could make the argument that, Rebecca talks about the institutional biosafety committees, and if all the research was done in institutions and that had those, and they all made informed decisions and did things, then maybe there would be a way to stop something that would be bad before it got further. But that's making the assumption it's done in public universities or institutions that have IBCs, that those IBCs are making informed decisions, that those scientists abide by those decisions and things like that. Those don't have the same kind of force of law as regulators do. But by the time it gets to the regulators, it may be too late. And as we said, those I think are more in silos and probably may not cover the risks that might be the most important ones for whatever the product or technology is.
Just going to add one quick thing, because the comment too was that, we're going to just follow a biosafety level, right? Which is containment for biological organisms, generally based on a risk assessment that it's infectious or, it's a pathogen. And we're talking about a potential risk assessment that's quite different than just knowing whether it's infectious or not. It's asking those questions. Ben, you mentioned, Rebecca, all of you have mentioned, like, what else can this weird, Richard, you said this weird, a thing that it can do. How do you do that assessment? Anyway, I asked that question because I struggle with this. Is like, if I just tell someone, do it at a BSL-2 and you're going to be okay, right? I don't think that answers or addresses all the risks. So that's just my comment.
I guess I would just sort of comment that I think again, focusing on the mirror bacteria is a special situation. I think one of the risks is once it's created, if you ask me, could somebody do it safely? If it was done in a single academic lab to build a single extremely biocontained, incapacitated organism that had all kinds of auxotrophies and so on, I think that could probably be done with minimal risk. The problem, and it's discussed in our technical report and in the science article, is once it's created, it becomes much easier to modify it. That might be done by a commercial actor who says, well, I need a more efficient platform, I need a more robust organism. Are those falling, are they going to be captured by existing laws? And then there's the dual use or malicious actor thing. And so again, in the context of mirror bacteria, we've suggested pushing things back to keep it at a distant horizon from the ability to create mirror bacteria. And that might involve, for instance, monitoring the purchase of reverse nucleotides and so on. I'm far from an expert about the practical ways to do it, but one positive note, though, is that the labs that we're aware of that were working on that, including John Glass and Kate Adamala on your committee, recognize this danger and immediately realize they should desist from pushing that boundary forward.
I mean, another consideration is we've had a pretty significant shift from a regulatory policy regime since this past January, and I would argue even a bit before that. I mean, on one end, the repeal of the Chevron doctrine, we're not really sure yet in the risk community what the implications of that are going to be in terms of deference to experts and panels within our regulatory bodies, right? So that is a fairly seismic shift in the risk policy and regulatory community that we're going to need to deal with. What does this all mean is the reason why I completely agree with Greg. It's questionable whether or not the existing institutional structures would be able to capture what's coming is because from a hard law perspective, they don't necessarily have the mandate to do that. Yeah, they might. It is questionable, and any time right now where you say it's questionable, if it goes to court, there's not, which is what you're likely going to see with the repeal of Chevron doctrine, at least in certain cases. There is going to be uncertainty relative to whether the interpretation of some of your regulatory agencies is able to hold up or not. So what would again, a new governance or new regulatory body, or not regulatory body, but like a new system or new group, what would they do? Essentially fill in the gaps with clear authority to deal with, again, some issues related to the convergence of emerging biotechnologies and machine learning and deep learning. Number one, and number two is deal with some of these process-based risk issues at a coordination level. Again, the tricky bit is you certainly don't want to make redundant or replace the well-functioning capabilities of our individual agencies, but capturing those margin issues is something that even though it's very difficult to do that through hard law, I think that's probably a necessary step in order to be productive in our innovation and engineer in good risk assessment early in the TRL process.
Thank you. Jason, you have your hand up. Please go ahead.
Yeah, thanks. Thanks to the speakers, and nice to see some of you who I've worked with before. I want to have us take a little bit of a step back. I'm thinking about an analogy. I'm thinking about how, in general, the biotechnology research community had a lot of confidence that their risk assessment and risk management was pretty robust. And then along came Gene Drive. And all of a sudden there was this sort of shift in thinking about, oh my gosh, there's something that we can do using tools that we've been using, but in a new way that might create a really different kind of a set of risks that we don't exactly know how to manage. And it seems like the same thing happened with Mirror Life, where even the previous discussions today, there's a lot of confidence in the synthetic cell community that we can manage these risks. Oh, except for this one thing, Mirror Life. And what I wanted to ask the panelists to think about is how, as a committee who's advising the research community about synthetic cell research, what can we promote that helps us identify where there might be another mirror cell field within synthetic cells? What's the process where we recognize something like that, where we deviate from the kind of stepwise risk assessment and ask something more fundamental about a new kind of hazard or a new kind of risk that no one quite foresaw?
I would say in any report you write, giving that as an example, as well as mirror bacteria, and again, emphasizing the importance of thoughtful communication. It doesn't necessarily solve all the problems, but I think that's a really interesting example. And I have to say, hearing some of the applications earlier today, before this, in the earlier panels, I don't want to say any of them scared me, but a lot of them had me thinking, I wonder what could potentially be a pitfall. And even hearing one of the speakers say, well, even something that's not replicating but is alive, maybe it could cause trouble. That was very humbling, because I have thought mostly about the importance of self-replicating and self-perpetuating, but I think your question strikes to the difficulty, and I think emphasizing to policymakers and scientists and the community that these things require a lot of thought and people looking ahead as far as our brains can take us, maybe eventually with AI helping, but certainly I think the thoughtful scientists are going to be incredibly important at all stages of this.
I think it comes down to diverse voices, right? With a lot of different expertise you might not normally have involved in that conversation, especially looking at things from a dual use perspective, right? And thinking about just not the direct application of a technology, but the roadmap that that technology could create and how it could be misused later on, and safeguarding some of those things associated with it, right? So the more voices you have, the better. It can't just be the synthetic cell community talking amongst themselves, right? It has to be a broad stakeholders and as transparent as possible to really have these really thoughtful conversations about how technology could go or how it could be misused.
I ditto what Rebecca said. You need to bring in other disciplines, other scientific disciplines, if you really want to get a broader view of what could be the impact.
What I'd recommend, again, Rebecca always has the answers, right? And they're always the correct answers. So I'm gonna stand by that. It's, if you're looking for a fun exercise, right? It's line up microbial risk assessment, chemical risk assessment, like all of your best practices going back 50 years, and look to see what are the common hazards that they tend to deal with, right? What common exercises have we been doing for a half a century or even more? And then ask yourself the question of, where are the current gaps, right? Just based on not science fiction, but where we think the field might be going within 5 years. Where are there honest gaps, right? Some honest hazards, or at least potential concerns that might occur, right? If you list all of those out on a separate line and then compare back to all of your different risk assessment capabilities, and you find something that is truly novel, right? That's where you invest your time. Right now, our risk assessment tools in terms of hazard characterization are very good, right? They've done an awful lot of work for a great different many chemicals, biological agents, et cetera, for a very, very long time. That doesn't mean that there aren't novel concerns that we do have, but it's something where, like, when I mean novel, it's you're looking at a hazard element that fundamentally changes the the physical expression of whatever object you're entering into the environment or to a certain media, right? Is there something that very new that we haven't seen before? I think that if you could tally a list of those in your report, or at least come up with a process to filter, to identify and then filter down some of these elements, that won't get you at your answer, but it certainly gets you further down the line, to focusing in on what's truly novel in this field versus what are the systems that might currently work that we just need to adapt, to be up with the times.
Small challenge there, Ben. Maybe one really quick follow-up, and then I see that Chris in the committee has a question. So, picking up on this idea of diverse voices and really trying to bring that in, I guess, does the panel have any practical suggestions? So, where does this happen in terms of like when? And I know the answer might be everything all the time all at once, but where does this happen in terms of who should be participating? And then what kinds of governance processes do these conversations fit into? Do we have them already? Or again, is this a case of setting up an additional kind of layer of governance? Is it informal? So just your thoughts there on, if we are going to encourage this, where do we start?
Just a very quick input is, this is probably not answering your question, but just be very aware of the development that's happening in East and Southeast Asia. Again, very forward-leaning, and getting back to, again, perception of risk and what is allowable, what is not. Again, very different risk culture and risk governance regime. And you certainly are going to see the field push the envelope globally, right? You're going to see different applications arise at different times, right? So it's when you're looking for different voices, again, you don't necessarily have to agree with any other country's regulatory regime, but certainly at least be aware of what's happening, what incentives are in the field that are driving development forward. They could be institutional, they could be economic, they could be academic, who knows, right? But so understand those incentives, try and make sense of why the field is moving or it's stymied in certain countries versus others, and figure out what lessons can we generalize from them, right? So it's not just up to the U.S., right, or us to look internally for lessons into biotechnology. Can we learn from the successes and failures of our partners and collaborators overseas as well? In East and Southeast Asia is a major hotbed for biotechnology advancement in that respect.
Emma, the only response I have to your question or hypothetical is, I want to believe that it's happening informally, and I think it is probably for and don't take this the wrong way, for good actors, and it doesn't mean other people are necessarily bad actors, but that there are some people who, as they're doing their research or as they're thinking of developing things, know and understand the need to bring in others to have conversations, or realize as they're going through it that, gee, maybe I need to have an ecological point of view here because I'm going to be releasing this, or I need to have a microbiological point of view, or a plant pathology point of view because of what I'm doing and where I see things. And so they bring those in, and I think IBCs and universities and lots of institutions do that and allow to do that. But I think there are probably others that don't necessarily do that, some because of lack of knowledge or other priorities or other things. So while I think it's probably happening informally on some level for many people, it doesn't happen consistently, and we're worried about the one that gets through that doesn't happen that way, whether that's a different country, as Ben says, or somebody in your own country. And so that's where the lawyer or the regulator in me feels like you have to set up some more formal institution to do it, for fear that, we're only as good as our last mistake. And that, unfortunately, you have 99 wins and you have one loss, but that loss can overpower all those rest of those wins. So I think, that's my concern in that area.
I wonder if setting up a series of red lines, right? Where, okay, if once the technology gets to this point, we need to reevaluate, right? But understanding where those lines would be drawn is very, very difficult, and it's hard to get consensus in the community. I mean, even with the Mirror Life conversations, do we make a synthetic ribosome or don't we? Is that okay? Is that not okay? Right? But understanding, I think, what we do, what based upon available information we don't think is risky, but then as things change, continuously reevaluating, because a risk assessment is truly a living thing. And it's going to change upon the amount of information that you get. And if there's some way to create that forced reevaluation on a more time, but it's not necessarily based on like years, it's based upon scientific development.
Thank you. Chris, I know you had your hand up a moment ago. Did you still have a question?
You and I have similar minds, and you asked a very similar question to where I wanted to go with the committee. I will ask, as I note that our time is cutting short. So following on this consideration, we've seen some, regulatory changes, some might even call it shifting goalposts for where we go with certain biotechnologies for our country. And even our committee itself has changed its overall topic as we continue to develop. What advice do you have for us as we navigate this risky world of regulation for what kinds of data you think would be best to be able to say, here's how we would do things like define the red line. Here's how we would differentiate between process and product. Here's how we would actually demonstrate a developer's intended use versus other potential uses. What sorts of things would you urge the ultimate decision makers who are not our committee to actually put in these kinds of regulatory hurdles for us going forward?
Okay. That's a tough one. Short answer is, even though I think it's been a year now, the number one thing that I would recommend right now is we need to figure out post Chevron doctrine what the boundaries of our institutions actually are. I'm not completely sure that we've solved that debate or really even begun to address that debate. There's been an awful lot of regulatory and administrative change, certainly over the past 12 months. Before we can make an educated evaluation of what's missing and where do we go, we need to first understand who has hard law statutory authority over different elements of the problems that we're raising. When do those responsibilities end, and what are they mandated to do? If that exercise isn't conducted, what we're building upon is something that, while it might be helpful, it could be pulled away at any moment because it's not necessarily backed up by the hard law written in requirements of one or more agencies. That is a pithy answer, but I think it's one that, again, if you're going to build a house, build it on good foundations, and this is the foundation that you need.
Speaking of foundations, you got to have clear definitions to try and remove the subjectiveness of the conversation, right? Some of us had to deal with a variety of regulatory changes in a very short period of time recently. And one of the underlying pieces was different opinions of regulations and different opinions of definitions and risk. And that is a very subjective conversation because every single one of us have a different tolerance of risk for a variety of different reasons, right? So how do you cut through that to take the subjectiveness away and have very clear definitions that people are going to interpret the same way and implement the same way and utilize the same way?
I would agree with Rebecca on the clear definitions. I think, you want to do whatever the best science is out there, but have the flexibility to change that as it moves along and the science changes and as we learn more. I guess I'm of the view that, Ben, that we're not going to find the answers to the post-Chevron thing for many, many years and many, many court decisions. And so I think we should just go ahead with business as usual and sort of worry about that later down the line. because there will be lots of things that still get regulated and lots of products that won't be challenged, and we will ensure safety of. And I think that in all cases, we are going to be putting square pegs into round holes. None of these laws were written for any of these technologies. And so if you take a very strict construction of Dobbs and post Chevron and things like that, then yeah, nobody's going to have any authority to do anything. But I don't think the public's going to be satisfied with all kinds of risks and all kinds of things going on and the government not getting engaged in that. And so I guess I'm of the view that agencies will continue to do what they're doing. Over time, we'll figure those things out, that there will be some middle ground that will come up, that is practical and rational and does a path forward. So I therefore think stick with the science but have flexibility in it to modify it as we learn more. To tag on then, who needs to do the risk science? Because in many instances, it's currently the person actually developing the product who says, here's how I intend it to be. This is what I think it should be. And here's all the data demonstrating safety. Is that good enough going forward for synthetic cells?
The answer is probably.
I was gonna say, I think that's the start, but not the everything. I mean, our system has always been set up with the developer and others to be the one who proves the safety and does that risk assessment. But you're going to need checks and balances with us, academic scientists, regulators, and others.
There's been a call for numerous, probably the last 5 years or so now, about funding applied biosafety research to actually put hard data behind a lot of the practices that we ask scientists to do. And thus far, there's only been philanthropy that's really done any of that funding. And the US government hasn't really stepped up to do any of that. But I think that would definitely help allow us put more confidence behind the procedures that we're asking people to do, as well as know, does that procedure or does that thing actually mitigate the risk that we think that it does?
All right, we're coming up on time here for our session. If any of our panelists have any final thoughts, I will leave you a couple of minutes to do that. And just to say, you set us a formidable challenge, especially at the end here, thinking about funding and definitions, both of which, even the conversations that we've heard today seem it's very difficult to pin down a definition of synthetic cells, for example. And so I think the, being able to structure sort of risk assessment processes to deal with the variety of purposes and types of technologies we're going to be coming up with. We're still going to be having to really think through what some of those key questions are, and really appreciate your guidance here today in this session on how to begin thinking through some of that. David, it looks like, do you have a final kind of quick question or comment for the panel before we close?
Yeah, maybe they could send their ideas. Ben mentioned that he could send a paper, but I'm also interested in existing regulations that we should look to as guidance. If there are any, what would you recommend? That's my question.
So it'd be wonderful if you have any suggestions for things that we should be looking at as a committee, if you would send those through to us in the coming days and weeks, that would be really appreciated. And I think that brings our session to a close. So thank you so much for your participation, to our panelists, to those of you who have been watching online. And I think now we are moving into some closing remarks from our study co-chairs, Felicia and Pete.
So I can say that for myself, I've really appreciated the inputs that we've heard today, the presentations, really engaging discussions. I find myself towards the end of this day saying, "Oh boy, is my brain full," and I need to spend some time percolating on what we've talked about and bring my best and freshest brain back when we convene tomorrow afternoon for a series of different discussions. So for those of you who, like me, are feeling like you've been challenged and your brain is full, please bring your freshest resources to bear tomorrow.
I would like to add to what Pete said. I have exactly the same impressions. It was a wonderful chance to learn so much. Thank you all, to all of our fantastic speakers. You have shared so much useful information and insights with our committee. And thank you to our audience for participating, listening in, asking your questions. We would love to hear your thoughts as well. And tomorrow we are going to be holding a closed session of this committee, and we will have the chance to talk about many of the items that were discussed today. So again, many thanks to our speakers for loading us with lots of information that now we have an evening to digest before we talk about it tomorrow. And also thank you to the committee. Thank you for staying with us, for your great questions, and we'll see you all tomorrow.
Bye, everybody. Thanks.
Goodbye.